Are we live, Control Center?
Yes, we are.
Thank you very much, dear CARIM community and all the other people that attend this wonderful session. I would like to welcome you to our annual CARIM Symposium 2021. And unfortunately, it's again in a downsized hybrid shape. We had these hopes high that we could all convene here together. Unfortunately, this is not happening, and so we're looking out for next year with full hope. Let's also think about our health professionals that cannot be here and probably not even online either because they have to work in the hospital saving all these patients from this horrible disease that has landed upon us in the last 2 years. But for now, we're going to keep the high spirits up here. It's very nice that at least a limited amount of people we can join here together and look at this very nice, a little bit shortened, but still very interesting program. CARIM, we have a prayer. Actually, we call it a mission, but we can also call it a prayer, so pray with me. CARIM, this is getting better every year. CARIM aims at defining and addressing key cardiovascular questions and clinical needs through application of efficient combination of basic and clinical researches and technology in an optimal team setting by combining expertise, track, translation and innovation and disseminate results to scientific journals, communities and society in general to contribute to early recognition and prevention of cardiovascular disease and improved cardiovascular care. This is one sentence and we can do it. And actually we're already doing it. And it's not only early recognition of disease that makes CARIM great and very important and a top institute in Europe, but it's also early recognition and identification of talents, which we are very good in. We have, since a couple of years, our very innovative HS-BAFTA program. And I believe that a lot of other schools and also faculties are a little bit jealous of us that we do this in this way. So we have for all stages of early academic career, we have a paid grants for disrupting or like not disrupting, but doing research during your master and bachelor phase, but also to prepare for a PhD. And also during our PhDs, we have short visits to international renowned institutes, where you can spend some time as a PhD student to get some cultural and research exchange and see how other labs do it, and then you bring that experience home with you. Makes you a more mature scientist. And then we also have a fully paid postdoc, which is also very new and very well made use of. Last year's grants were given to Danique Meijs to prepare for her PhD. The PhD short visits were done by Kim Maas, Jordi Kokken and Job Stoks. And as a postdoc, Jens Posma, who's probably now in Mainz and not here. Are you here, Jens? No, he's not here. So he'll probably look from Germany to this meeting. We'll come back to this later in our prizes and award session. But we also need to attract talent from outside. This is also one of the recommendations of the External Review Committee. And we also did that successfully. And you can see here all the people, talents that we got from outside, young toward a little bit matured or aged talents, how should I call them? But this is very good. And actually, CARIM Maastricht is now internationally also on the map because these people all want to come work with us, and which is an exceptional merit to us CARIM as well. So Ed Eriga joined us, Bastiaan de Galan, Mark Hemelder, Andy Baker since very shortly. Welcome Andy also here in the audience. Naza Hamdani, she's actually from the University of Bochum, and as you know that Martin Paul, our previous president, will move to Bochum, so we actually call this the prisoner exchange. So Naza will come to us and Martin will go to Bochum, but also they will still interact because part of the time Naza will stay in Bochum as well. Bastiaan Boukens, we actually decided that yesterday. That was like the final approval and his decision to come here as well. And of course, Jurten Berg also this year joined us as a professor. We also create our own talents. We have many programs for that. We have the faculty talent class, which this year Dominique Liens, Miranda Schramme and Ingrid Dijkgraaf joined. It's a very prestigious program, and this leads to a professorship within 5 years. We have our tenure track, which probably will be changed in a career track, because as we know, the new CEO regulations tell us that we have to give a permanent position already after 16 months. So the tenure track is a little bit unusual in our shape, but this is still very good that these people joined this tenure track. Thomas van Sloten, Matthijs Kluitmans, Pieter Goossens, and also recently Bart Spronk. Good luck to them all with their academic career within CARIM and Maastricht. We also have an FHML, our faculty tenure track, in which Martina Calore and Miranda Nabben are enrolled. And if you do a quick calculation, you can see it's very nicely divided over the sexes, 5 to 4, 4 to 5, how shall we call it? We all know that CARIM is very easily built up of the 3 major divisions: blood, vessels, heart. It's easy, everybody can understand that. This is why we are really also well known throughout the world. There are many cardiovascular institutes that have the heart and the vessels, but combining it with blood, that is a unique feature of CARIM, and this is why we are doing so well in international literature as well. You know our website. If you don't, please visit the website. It's very nicely, it's new. I have some figures from 2020. They're also in the annual report. There's still some copies outside at the coffee. If you want to pick one up, you're welcome to do so. We're pretty steady on about 20 million of annual budget. Last year we had 9.4 million euros of new contracts and grants. And since a long time we already have 11 clinical departments and 6 basic departments within our institute. Also telling you already that we are very translational. We're trying to get the clinicians into the basic departments and vice versa. And this is really one of the strong features that CARIM has. Last year we produced 36 PhD theses, which is a bit low, but this is mainly caused because of all the online defenses that people didn't like, and they just postponed it to the next year. And this is why we already have more than 50 now, Wouter, correct me if I'm wrong, more than 50 already now, so this will be a top year for us for PhDs. We have 156 full-time equivalents of researchers, supported by 48 FTE of technical supporting staff. And last year we had a record of 660 SCI publications with an average impact factor of 6.1. And Dora agrees with this because this is not an individual figure, this is a figure for institute, so don't worry, this is Dora compatible, this average impact factor. What about grants and contracts? As you can see here, this is sort of the last 6 years. Something went wrong in 2016, I believe, Wouter. 505, that's not possible, I think. So this is shown here in kilo euros. And as you can see that in 2021, in September, we're a bit low. But this is actually the same situation that we had last year. And we're always pretty efficient in getting some more of these contracts in the last couple of months. Last week we had a very interesting week because it was the ITN deadline, the Horizon should be not Horizon, Europe applications in 2021. We from biochemistry did too, but CARIM as a whole did a lot of these applications. You can see here, this is the faculty application list, and in green is CARIM. So we did more than 2/3 of all applications came from CARIM. Considering that we have 5 or 6 schools and 2 institutes, this is a really, really good effort and really good. So if we get them all, we have 4.5 million euros, so that will make up for the loss that we had a little bit during this year. We had a couple of large contracts. Zon Doelmatigheid, LEAP application by Justin Luurmans that was granted. A very large Abbott research agreement for Frits Prinsen here in the audience. Congratulations, Frits, on that. And also another Horizon 2020 Rita MI2 from Arnoud van 't Hof for 615 kilo euros. So this is very welcome, of course, because a lot of work can be done with these things. And you can see There's a lot of clinical work also, but there's also extending to the basic departments within CARIM. We had some very prestigious personal grants, and those people you will hear later also in our program. An NWO Rubicon for Stepan Denisov, who will go to the UK with that grant. An NWO Vidi for Jordi Heijman. Unfortunately, Jordi fell ill this morning, and his temperature is that high that he even cannot present online, so we have a little gap in the program. So good news for all the speakers, so they can be a little bit relaxed about their 15 minutes. And we have an NWO Aspasia for Vanessa van Empel. A Dekker postdoc, which is also very prestigious, for Uyen again, and she's also here present today and giving a presentation later. And also a Marie Curie postdoc fellowship for Dizar Kader, and he is also here present today, fortunately. Some actions that we did last year and the year before. We thought it was very important that we should give clinicians this safe day of research activity, so 0.2 FTE, 1 day in the week, and go away from your clinical department and come sit with the basic departments at the UNS 50, so that your research day is protected. We actually gave 12 clinicians 1 day in the week of research appointment, bringing the clinicians that have research appointment at CARIM to 34 in total. We have then also decided that to give some support staff that had a longer time already on these loose contracts, to give them a permanent position, and 6 of these support staff people were now given this fixed position at CARIM. We also decided to hire some more support staff because we were a little low, and some groups actually, because support staff left, were void of any support staff, so we gave them new support staff to help them with the research. And also, as you know, and also Judith will come back to that later a little bit, is that we invested in almost $1 million in equipment. And that was flow cytometry, single-cell sequencing, and circular dichorism protein structure analysis. And this is now all here, it's operational, and everybody can use this equipment powered by CARIM. We also changed our organogram a little bit, and I'll explain this if I can find the pointer. We have an executive board known to you as the DB, the daily board, in which we are advised from the Education Program Committee, the Research Council, and the Strategic Board. And in the Strategic Board, we will have CARIM PhD student now, which is new. We have one of our support staff members in the Strategic Board, which is new. And then, of course, this is what you know, the 3 divisions with our 6 programs together with the departments in the School Council. And here you can see another addition to the Executive Board, the Daily Board of CARIM, which are called the CARIM Priori, but I'll come back to that at the last part of our program. We then have some highlights. We all know Shahinda Hossein. She will also present today, but she's now in Paris presenting some other interesting grant proposal there at the embassy. And so she decided that that would be better. And also we, with this condensed program, said, well, there's room for you to leave, so do the good thing and get more money in Paris. And we will show your vernissage. So this is a very interesting piece of art. This is a, you can see it, floating placenta. It's in alcohol, and it's very interesting. It's really floating, so now it's not a movie, but it's still, so you can't see it. And this was in Eindhoven a month ago, and together with her friend Kiki van Eyck, they made this very nice lithography that you can see here. And this is, showing the Queen of Hearts. This is very nice. This is lace, and this is a very nice impression of the heart, and also the microvasculature in the heart by this lace. So I think it's really cool. We bought one from CARIM, so you can within short times you can see it hanging in our war room. And actually, she's still active in crowdfunding. And this year, our CARIM crowdfunding will go to Shahinda Hossein for her research called Queen of Hearts. So she doesn't know this yet, so if she's online, she will like to hear this. Another highlight is Kevin Vernooy. You all know him, our new young professor with many hats. So you see him here with one hat, but he has more hats. He is also PI in CARIM. He's head of cardiology. He's running this department really well. And he's also chair of the division heart. So welcome also to you, Kevin, here today and in our CARIM staff community. We almost all participated in the current version of Walking with Doctors. As you can see here, still a lot of money was collected for the Health Foundation Limburg. And also from that foundation, many interesting projects are being performed. And later on, you can see the prize winner of today's or this year's first round of the Harry Krijns Grant Award that is funded by HFL and also supported by, and I'll read from my card, Bayer, Amgen, and Sanofi. So thank you very much, sponsors, for that. We also had our World Thrombosis Day, and as I am never invited for Op 1 or GNEC, so I have to make my own show. And you can see it, you can effectively do that at our university, so you can rent a studio. And then we have these very nice presentations this year by Martijn Smulders, Eline Kooi, and Arnoud van het Hof. And it was about myocardial infarction. It's an interesting program. We do this every year. So, and also next year, some of you will be asked to perform also in this World Thrombosis Day every year at 13th of October to present and to get used to contact with the general public. And now we do it online, but hopefully next year we can do it on site again. Some of our top citation scholars, we were informed that both Koen Steehouwer and Christian Weber belong to the top 0.1% of citations in their field. And there are probably some more also here in this audience. So please, when you get notice of this, forward it to us, and then you'll also be on the big screen at our next annual symposium. So congratulations to Koen and Christian. We're very proud of you, and thank you for doing this for CARIM. Focus, future, the iPSC lab, many of you will have heard about it. It's ready. It will be opened on 8 December, and we think it's a very strong future line within CARIM by recreating patients, especially now because our animal experimentation is a little bit, what shall we say, unsecure. So by recreating patients in the lab through differentiated iPSC cell lines, we can actually recreate the patient in the lab. And we can study those patients, and we can also test therapy. We could also, of course, study transplantation, but I think it's the best for modeling therapy by gene modification, also translation modification, which Elisabetta Castoldi is an expert in, and also to test drugs. And personalized medicine with this kind of technology will come within reach in CARIM. So unfortunately, we cannot invite too many people. Leon will coordinate this together with Cengiz, and 8 December we'll try to get together in this new lab. It's on the second floor in the middle lobe. Probably you will have passed it. It's state of the art. It's very nice. What other things are we doing for CARIM? So as already said, we're going to structurally extend our support staff with 2.0 full-time equivalent. We will give each division the opportunity to appoint a 2-year postdoc based on 50-50 financing, 50% from CARIM and 50% for the division. So any strategic goal you would like to reach, you can put a postdoc on that for 2 years. And then also help to restore this middle layer that CARIM is actually lacking a little bit. We're also going to invest in cardiovascular data science. Jordi Heijman is very active in this community. And we also, from CARIM, have made available some money for personnel and also for some training and coordinating this new initiative. And then for also to still prevail in our diversity and inclusivity program. We do 10,000 per year for courses, for lecturers, and some other stuff to get this going also within CARIM. And as last, we also, as a result of our last school council, in which Professor Schurgers made a double appointment, he actually had to lecture in Papendal, but he also had a lecture with our school council. And then we started chatting about just stuff. And the animal center was one of those topics, and we said, well, maybe we should do this more regularly. And then Wouter approved the idea that every first Monday of every month we can have some drinks and chats in Saam. First, we have to start. I really apologize, PhD students. It's horribly unfair, but we start with our permanent staff because there's only room for 50 people. It's like a separate area in the bar. If now for any reason there's not too many people, like maybe 25, then each next time staff members can introduce somebody which can be anything, a dog or a PhD student, to join them to the CARIM Café. And this will actually start next month on December 6th, Corona Volente. Then finally, today's program with a very nice front. You can see CARIM is lifting up the heavy load of what happened to CARIM the past years, but a very strong pose of this scientist looking forwards with a positive mode, I believe. And you can see the program of today. We will now give the microphone to Judith Sluimer, who will moderate the first session on talent career development grants, in which we will have 3 presentations instead of 4. Then we perhaps have a little bit extended break afterwards, and have an interesting afternoon program. Roger Veltrop will share his experiences with his crowdfunding and his, I should say, incredibly interesting life, although it has some downsides, but you will tell us about that, Roger, later. And also then the secret winner of the Harry Krijns Research Grant will be unveiled at the 3:15. Then we have an online Reneman lecture. Unfortunately, Barbara Cassadij, she will tell us on radical views on atrial fibrillation, and this session will be moderated by Paula da Costa. And then we get the CARIM Spotlight session, and you'll see what that means later. So with that, Judith, please step up.
Well, thanks, Tilman, for that nice overview, I would say. Always good to have an inspirational talk about how great we are, right? Thankfully, it's backed up by data, as scientists like. Are you going to leave that one there? Thank you for the students. So before I'm going to introduce all our talents, I would like to share with you, already alluded to by Tilman, one of the acquisitions of CARIM. Of course, it's not the only one. Christian Wouters was very important to also purchase a flow cytometer, a very special one, the Cytec Aurora. And if you haven't started using it, you should really talk to Christian about that. But of course, my passion is really about the single-cell sequencing, so I'll take the opportunity to share that with you. We've installed this controller. It's going to be only operational until mid-December because we're missing some plastics. You've all experienced this in your lab, I suppose, and unfortunately we're not the exempt. But more importantly, CARIM really wants to spur research in this area, and we really think this will help you get more grants and hopefully get more, even more acquisition numbers for Wouter on the next, in the next table. So what it is that you can make a small proposal, half a page, send it to the CARIM Secretariat, and then there is a committee including Leon de Wind, Leon Schurgers, Monica Stoll, and myself, who will help you make this into a viable proposal. So it's not really to judge, it's more really to help you make the best decisions in your research and to guide you through the different opportunities there are with single-cell sequencing or single nuclei sequencing or ATAC-seq, whatever question you have that you want answered. And CARIM measures 50% of the reagents, not the sequencing, but 50% of the reagents. And that's actually already quite an important step, of course, to make this lower threshold. So please submit your calls, and let's see if we can get this into the CARIM ingrained in this new technology. And also to lower this threshold, and if you're not so familiar with single-cell sequencing, we're going to organize a workshop in the first quarter of the next year. The first one will be about the theory, about really hands-on the practical steps in the lab with the help of 10x, and then later we're also going to organize a bioinformatics workshop, because we know this is also definitely a bottleneck for a lot of people. I mean, it is possible. I learned R in the first lockdown, and I can do it, but it takes me forever, so I also need help. But still, it is definitely something that we can all learn if you want to. But that's that for the 10x. So I'm going to open the next presentation in the meantime. Oh dear, this is a Mac. I'm getting out of this, I hope, right now. There you go. So CARIM already showed there's quite a few talented people in Tilman already showed all the talented people in CARIM. Yes, there we go. So the first one that is going to present is Stepan Denisov. And Stepan is actually one of our international people. He started working in Moscow, then came to Maastricht and did his PhD with Ingrid Dijkgraaf on the tick proteins in the biochemistry department. And now he's actually moving across the channel to Oxford to even study more in-depth protein biology. And he is going to share with us his Rubicon project. So there you go. Welcome, Stefan.
Yep. Thank you for introduction, and thank you for inviting me here. So after 7 years at CARIM, I've finally made a CARIM symposium. I'm indeed working with Ingrid and Tilman on chemokine binding peptides and proteins. So that's already been told, so that's a short slide about my glorious career development. And I'm indeed going to move to Oxford. But this presentation will be about what happens at Maastricht for the last 7 years, which is terrifying 25% of my life. My PhD was focused on chemokines and chemokine binding proteins. So chemokines are a really small protein, a secreted protein which regulates cell trafficking. But despite they're really small, around 80 amino acids, they have a lot of activity. So they bind glycosamine glycans on a cell wall. They can dimerize and oligomerize and even heteromerize. And they, of course, activate receptor on cell membrane to regulate cell trafficking. To make situation even worse, humans have 46 chemokines and 23 chemokine receptor, which can interact with each other. So that makes the system really complicated. And despite that, chemokine signaling is involved in a lot of physiological and pathophysiological conditions. We have right now only 3 approved drugs which targeted chemokine receptor. So that means that we don't really know how to intervene chemokine signaling. In contrast to humans, pathogens, for example, ticks, can do it quite well. So, for example, ticks express special proteins called evasins, which successfully bind chemokines and basically block chemokine signaling and, for example, inflammation. So evasins also quite small, around 100 amino acids. They disulfide-rich protein, so that makes it very stable because they have to survive in bloodstream. Ticks have around several hundreds of evasins right now because, well, ticks a little bit genetic freaks, so they have around 4,000 proteins in their saliva. And my main focus was on 2 proteins, evasin 3 and evasin 4. They bind different chemokines. Evasin 3 binds 6C type, for example, 6L1 and 6L8, whereas evasin 4, it's a binder of CC type chemokines. And I'm chemist by training, so I really like to consider proteins as molecules. So we started with elucidation of structure of these proteins. And the problem with evasins is that they have too many disulfides. So, we need to figure out how they connected to form a native structure. So there are a lot of basically ways how you can decipher disulfide connectivity. But if you have a hammer, everything is a nail, and we have NMR spectroscopy, so we developed our own method how to do it. The main challenge that sulfur is not visible in NMR, if we can say it like that. So we developed a method when we can replace sulfur by selenium, and that makes a lot of changes, selective changes in NMR spectra, and we can decipher disulfide connectivity by just visual inspection. For example, we can solve disulfide connectivity of very small peptides like conotoxins here with 6 cysteines. So we've done it for evasin 3, and turned out that evasin 3 has so-called inhibitory cysteine node, when 2 disulfides form kind of a cycle, and the third disulfide bond actually protrudes it. So it's very common fault for spider toxins, for example, but for tick proteins is not so common. And when we apply this, when we introduce this disulfide connectivity in a structure, we can solve it, and we can see that evasin 3 form beautiful clover leaf structure with a lot of disulfide inside core. So we solved, we also figured out how it binds chemokine exactly. And as you can see, evasin 3 binds beta-1 strand and alpha helix of monomer of CXCL8, but it doesn't affect receptor binding site, but still can inhibit chemokine activity. What is even more interesting, because, well, chemokines bind GAGs, evasin 3 disrupt GAG binding site of CXCL8, which you can see on the left. When it binds chemokine, it just disrupted and basically displaced it from GAGs. But if you move to CXCL1, another ligand for evasin 3, it will also disrupt this site. But on other side of CXCL1, there is additional a patch of positively charged residues, which also can bind GAGs. So we hypothesized that if you label evasin 3, so we can basically visualize CXCL1 deposit on GAGs. And by that, we can, for example, visualize place of inflammation. And that's indeed worked out. So, for example, we can visualize CXCL1 deposits on cells and even on arteries. So that was about evasin 3, but evasin 4, it's also called evasins, but they're completely different family. NMR was not really suitable for this protein, so we collaborated with a group of Bert Janssen from Utrecht University, and they solved it by X-ray. And as you can see, evasin 4 is completely different from completely different structural family, and it's arranged in way more complex way. So it contains 8 cysteines, there are several subdomains, a lot of beta strands. But we showed that main binding spot of evasin 4 for chemokines is actually located in flexible N-terminus. So we can take it out, and it still will bind CCL5, for example, and will possess anti-chemokine activity. And also it retains biological activity, maybe less active, but still potent. So main question of my work was like, could these proteins be drugs? And the most honest answer, of course, would be no. Because it's very difficult proteins to work, and mostly because they are foreign proteins, they from ticks, and our goals for therapeutics and goals of ticks are not really aligned. But I still believe that we can adjust properties of evasins or their parts to meet our needs. And that will be main goal of my Rubicon at Oxford. But of course, alternative way would be create chemokine binding peptides from scratches. So that's small glimpse of ongoing project with company called PepScan. And in this project, we attempted to create, obtain chemokine binding peptides by phage display. But of course, with such a character, so we cannot use just regular phage display, so we used mirror image. Oh, that's really low resolution. So basically, in contrast to regular phage display, in mirror image, as a bait, we used protein which created from D amino acids. By that, we can select L peptides, but in the end, we can just swap stereochemistry and we will gain D peptides, which of course more stable, less immunogenic than L peptides because, well, all proteins just recognize L amino acids. So we synthesized D6L8 by the scheme you see here and used a library of cyclic L peptides with 10 random positions. So after 3 rounds of selection against D6L8, we sequenced enriched library, and we had around 500 peptides. So you can see clusterization of these peptides on panel B. So there are several clusters, and to our delight, these clusters have some level of conservative residues, for example, hydrophobic residues in position 2 and 3. So 195 peptides from this library was synthesized, and we screened it by SPR against 6L8. And we have a lot of good binders, but because this binding still could be non-stereospecific, we also measured binding to L6L8 to make sure that, well, our L peptides still bind only D6L8, but not L6L8. And that, of course, decrease number of potential hit, but we still selected 5 peptides, so they depicted on panel E. And when we synthesized this peptide from L amino acids, we saw that it nicely binds D6L8, but not L6L8, with, in that case, KD around 200 nanomoles. But when we swap stereochemistry to D amino acids of these peptides, we saw really nice change in binding of 6L8. Now we have binder against L6L8, so what we have in nature. And of course, we are going to study it next, and we also have preliminary results about biological activity. But that's a really nice project, which I hope I will continue when I leave temporarily to Oxford. That was a short overview of my work, and I want to thank all people who worked with me and contributed to our papers and funding agency. So we have a bunch of grants from NWO and Coatstar Fellowship from Maastricht University and UMC+. And of course, I want to thank you for your attention.
Thanks a lot, Stefan. So before we start with questions in the audience, I would like to let you know that there's also a chance to ask questions in the chat. I've just opened the chat box here, so please send your questions into the chat. But for now, before they do that, any questions in the audience here for Stefan? Yes, Andy?
Now it's on. Finally, we got there. Congratulations on the work, and I'm sure you'll have great success in Schumer's lab. Have I got it right that the idea to use the peptides is because the native proteins from the tick will be immunogenic? if you give them to patients in a disease setting. Will the peptides also not have immunogenicity? I mean, we also tried to use phage display peptides to modify gene therapy vectors a little bit, but there were also peptide-mediated immune issues because of that. Is this you have great affinities, but are the peptides still going to be immunogenic if you use them in patients?
Yeah, of course, peptide also could be immunogenic. So that's, for example, in our last project, we tried to use D amino acids, which when D peptides less immunogenic. And that's one of the reasons why, in my opinion, we should migrate from entire protein to peptides, because on peptides we have more control, so we can easily synthesize it, we can easily modify it. So it's better than to have, for example, 110 amino acids protein, which probably should be expressed and then correctly refolded. So in that case, if we can, for example, take out only binding interface of these peptides and make it synthetically, so we also can modify it to adjust, reduce immunogenicity, adjust KD, and so on and so on. So in my opinion, for example, native tick proteins could be a nice start, but probably cannot be nice therapeutics in the end.
Thanks for that. And there's Tilman who has a question.
Actually, you should never be allowed to ask questions with your own people, but I'm sorry, Stefan. So the action of chemokines is very redundant. So what would be the best strategy? Get a redundant inhibitor system or selective so that you combine different sets of inhibitors to make an inhibition cocktail of choice?
Well, there is quite a debate about that. For example, I always thought that having selective chemokine inhibitor would be better because we can just make a cocktail from them and inhibit chemokines we need to not affect what we don't want to touch, for example. But some people, and among them my future supervisor, they thought they think different. So just to have redundant inhibitor would be better because we can just by one compound block entire system itself. What is better? To be honest, I don't know. I'm a humble chemist. I'm not really a biologist, so I cannot judge on that. So we will see. I hope at least one of these options would be a viable therapeutic in the future.
All right, thank you for that. Considering time, I think we should probably continue the next one. But thanks, Stefan, for a nice presentation. And okay, thanks. And I would like to welcome Uyen Nguyen to the stage while I set up her presentation. And here is our second international speaker of the day, talking about diversity, I would say. So she studied in Vietnam first, but then came to Maastricht also, not to Twente, to study technical medicine. So she is an MD, but with a very special background. I think that's very interesting, and I'm very happy to hear about the work she's going to do with the Becker postdoc fellowship. Uyen.
Well, thank you for the kind introduction. I'm indeed a resident cardiology and a clinical technologist in the center. And it's a great honor for me to present our DECKER project today about the relationship between cardiac resynchronization therapy and ventricular arrhythmias that will start in 2022. I have to admit I have some Asian looks and my roots are in Vietnam, but I was actually just born in the Netherlands. So that makes me a Dutch citizen. So I'm not a really international person in this room. So heart failure is a frequently occurring disease associated with high mortality and a high patient burden and societal burden. In about a quarter of the patients with heart failure is caused by an underlying conduction disturbance. In the left figure below, A patient with a conduction disease is illustrated, where you can appreciate that the electrical conduction propagates from one side to the other, and the total activation time is prolonged. In the right figure below, this patient is treated with cardiac resynchronization therapy, which is a type of pacemaker that involves 3 leads that creates more synchronous electrical activation by simultaneously pacing the left and right ventricle. CRT has been a highly successful therapy in the majority of patients and is recommended in heart failure patients with conduction diseases and current guidelines. In the Netherlands, only annually more than 2,000 implantations take place, and we observed that ventricular arrhythmias are present in 6% of these patients within 1 year after implantation. And this is a considerable substantial problem, considering that these ventricular arrhythmias can be life-threatening. For selection and evaluation of CRT patients and for a more in-depth understanding of the etiology of ventricular arrhythmias, evaluating the electrical activity of the heart is crucial. Currently, this is done by the standard 12-lead ECG, which is a very accessible and valuable clinical tool, but only represents an averaged electrical activity and lacks spatial resolution to detect local repolarization and activation abnormalities. ECGI, or known as electrical cardiographic imaging, can be interpreted as a more high-end ECG, involving the acquisition of the electrical activity of the heart with about 200 electrodes on the torso. With a heart torso geometry derived from CT and a mathematical model, the electrical activity of the heart can be reconstructed non-invasively. On the right side of the slide, you can appreciate a patient who undergoes an ECGI measurement in clinics in Maastricht, and on the figure on the top left, you can appreciate an ECGI map of the heart. In this map, every color represents a local epicardial activation time. So in this representation, early activation is represented by red, and late activation is represented by blue. And in this patient, the electrical activation propagates from the right side of the heart to the left side of the heart. The ECGI technique used in this project has been in-house developed and validated in animal studies by Dr. Kluitmans and researchers from the Department of Cardiology and Knowledge Engineering. And recently, one study by Dr. Kluitmans also showed that ECGI also provides us more insight in arrhythmogenesis of patients with idiopathic ventricular fibrillation. On the right figure, you can appreciate an ECGI map of a normal patient, a patient with frequent but innocent PVCs, and in a patient with idiopathic ventricular fibrillation. And you can appreciate that in a patient with idiopathic ventricular fibrillation, higher gradients of repolarization times were present, and PVCs arise from sites of early repolarization. And these two factors were probably primed idiopathic ventricular fibrillation. In my early years as a clinical resident at the Department of Cardiology, I was taking care of a female patient at the cardiology ward in Maastricht. The patient had a history of aortic valve replacement and was referred for CRT with a class 1A indication, which is the highest indication for CRT that a patient can get. She underwent a normal CRT implantation procedure, but was hospitalized at the intensive care unit within 2 weeks after implantation due to an electrical storm, which are episodes of life-threatening ventricular arrhythmias involving more than 30 ICD shocks. In this patient, I performed ECGI measurements together with a PhD student, Job Stocks, who also received a BAFTA this year. And the ECGI maps from this patient you can appreciate on the right side of the slide, and are color-coded for repolarization times. We observed that in these patients, the total repolarization time during CRT was shorter, but more heterogeneity of repolarization times was present. And although we cannot tell for sure whether this observation was an explanation for electrical storm, the conventional CRT therapy was discontinued, and she received an alternative approach for resynchronization, a His bundle pacemaker instead. and the patient did not suffer from any episodes of electrical storms ever since. This patient was my inspiration for this project. CRT apparently has a lot of positive effects, but also some negative effects. One could argue that by placing an electrical lead at the outside of the heart, so at the epicardium, a non-physiological activation wavefront is created, which leads to a more dispersed repolarization pattern and is an electrical substrate for CRT, for ventricular arrhythmias. On the other side, one could argue that by biventricular pacing, a more synchronous activation and a more synchronous repolarization pattern is created, which results in reverse remodeling of the failed heart and is therefore a more protective effect of CRT. Currently, we do not yet understand where in this range of positive and negative effects each individual patient is positioned. For this purpose, in the current DECAD project, I will investigate how conduction disturbances and subsequently how CRT influences local repolarization patterns on the heart, and will try to figure out whether these CRT-induced repolarization changes are associated with ventricular arrhythmias. For this project, I've defined 4 work packages. In the first project, we will simply compare local repolarization differences between patients with a normal electrical conduction and patients with electrical conduction disease. And on the right side, you can appreciate in preliminary data that I processed with my technical medicine student, Mark Smeets, that in patients with the conduction disturbances, an altered relationship between repolarization and depolarization is already present compared to patients with a normal conduction. In the second project, I will investigate the influence of CRT on local repolarization of the heart within the same patient by performing ECGI measurements prior and after CRT implantation. And in this project, we will also look at the trigger-substrate interactions, where the substrate are the repolarization patterns that we measure with ECGI, and the triggers are spontaneous PVCs that we catch during the ECGI measurements and the paced beats. In the third project, I will collaborate with the computational researchers in Lugano, in Switzerland, and will use the data from our Maastricht ECGI patients to reconstruct patient-specific computer models of the heart. In these models, we will virtually test different approaches of resynchronization and will try to find the most individual pacing approach that leads to the most advantageous activation and repolarization pattern. On the right side, on the right side, you can appreciate some preliminary computer simulations from Dr. Pezzuto and Dr. Verzal from Lugano and Maastricht. And in this virtual CRT patient, CRT led to a smaller histogram of repolarization times, which means that there is a more synchronous repolarization. And apparently this patient was responding to CRT, so apparently more synchronized repolarization precedes response to CRT. In the final project, I will collaborate with clinical researchers in Bordeaux, where they have a unique and large dataset of ECGI maps from CRT patients. Combined with the Maastricht ECGI cohort of CRT patients, we will accomplish one of the largest CRT ECGI cohorts. And in this project, I will investigate whether these repolarization patterns that we detect with ECGI are associated with the occurrence of ventricular arrhythmias by simply interrogating the device. In summary, in this DECAD project, we will investigate the relationship between CRT and ventricular arrhythmias by using a high-end ECG technique to study local repolarization changes of the heart. Secondly, by using computer modeling to virtually find the optimal pacing strategy of each individual patient. And finally, by conducting a clinical study with a large patient cohort by associating these local repolarization changes with ventricular arrhythmias. With this project, I hope to provide a more patient-specific risk assessment prior CRT implantation and a more individualized pacing strategy for each patient, hopefully avoiding unnecessary risks for pacing-related ventricular arrhythmias. I would like to thank you all for your attention, and I would like to thank the host institute CARIM and Maastricht University Medical Center, the Dutch Heart Foundation, and my collaborators from Lugano and Bordeaux. And I would like to particularly thank Frits, Kevin, Matthijs, and Angelo and Mark for supporting me with the writing of this proposal. and also my clinical supervisors, Richard and Chang, for giving me the opportunity to work on this proposal during my clinical duties as a resident. Thank you.
Thank you, Nguyen. Yeah, I think LinkedIn does not indeed contain your place of birth. I was confused by your bachelor in Malaysia, so I'm sorry about the assumptions. I should have not have made those. So I think it was a really nice talk. Is there anything in the chat yet? Not yet. Maybe there's questions in the audience. Yes, Hugo? While he's getting the microphone, so maybe some sort of inspirational question. Tilman already said that there's clinicians are being supported in to do research, and how does that work for you? And can you tell maybe some young MDs in the audience how you manage to combine your research with your clinics?
Yeah, that's a very good and personal question. It's quite, in my opinion, it was quite difficult in the beginning to combine research with clinics. And I think the first thing you have to do is accept that you can never be as good as these full-time researchers at CARIM. So that's one thing. And furthermore, really talk with your supervisors and your clinical supervisors about your future plans and your wishes for the future, and mostly they will understand, they will help you to accomplish that. That's my experience. But also just work in the evenings and weekends on a project instead of daytime.
Well, it seems you're doing quite well, and you've got a lot of support with all the people that are working there. But thanks for that question. And then Hugo?
Thank you, Jan, for a beautiful presentation. And, fantastic project. For a non-cardiologist, it's hard to come up with a question. Let me try. In the third part, you were looking at the patients, and you're distinguishing responders and non-responders with CRT. How large is the fraction of non-responders currently? Maybe you said so, but I missed it.
Yeah, it's about 1/3 of the patients who receive CRT.
1/3 of the patients. So those that are currently non-respondent are managed by medication, for instance, or other ways? Or
so CRT is an additional therapy next to optimal medical therapy. And when the patients who receive the CRT device do not respond, we will still continue with the optimal medical therapy. We will try to monitor them as good as possible at our outpatient clinic for heart failure patients. And but CRT is rarely discontinued in these patients. Because they already.
That your studies will also find clues to the fact that patients are non-responding.
I'm sorry.
I guess you hope that your studies will also find clues.
Yes, exactly.
Non-respondents.
Yeah. So most likely, although I did not present it here in this presentation, non-responders to CRT also have a higher risk of ventricular arrhythmias. And the question is whether this is caused by less reverse remodeling of the heart, or also the occurrence of these ventricular arrhythmias. But we also hope that we can use ECG imaging to better select patients for CRT and to better guide the implantation during CRT, so find the optimal place for instance, for the electrodes. And so that's also part of the project.
Thank you.
Yeah.
Okay, thank you. There's a question from online from Dr. Elham Bidar, who asks, would it be an idea to correlate the depolarization changes to underlying fibrosis patterns qualified by MRI? I don't know what that means. But,
that's... which doctor was it?
So Elham Bidar has asked, is it an idea to correlate the depolarization changes to underlying fibrosis patterns qualified by MRI?
Yeah, that's a very nice question actually, because now we also have the opportunity to perform ECGI with MRI scan. So we also have information about the location of myocardial scar. And in the first slide, I don't know if I can play this. Oh yeah, so this is an ECG map of a patient, and the green underneath is actually myocardial scar from MRI. So it's very interesting to combine this because we know that ventricular arrhythmias can also occur from sites of myocardial scar. So it's a substrate for the occurrence of ventricular arrhythmias. So we have this information, and we will also try to incorporate it in the trigger substrate investigation of ventricular arrhythmias of the CRT. Yeah. I'm looking at you because you. Yeah.
So I hope he's happy with the answer. Maybe you can put that in the chat. We have one more question, I think, from Harry. Someone has the microphone. Oh, I'm sorry, I didn't see that. Uli had.
Yeah, Ian, thank you very much. Beautiful presentation. For the parameterization of your model, of course, data on ionic current activities would be very useful, because only from the ECG parameters in itself, it's difficult to really constrain the model such that it can really realistically reflect the repolarization patterns and the changes that occur. And this, I think, stresses the need for actually basic research and also animal work essentially in that field. But my question to you is, are there any ways to get an idea on how you would have to change the currents in case you do see repolarization changes?
Oh, that's a very tough question. I actually don't know how I can... So how you can change the ion currents to reduce these repolarization changes? That's your question.
Right. I mean, if the idea is that the model reflects the repolarization changes, then of course you have to start to change repolarizing currents, and you need a basis for that. So it could be, for instance, that there is already knowledge on the effects of pacing at certain sites and the effect of that on repolarizing currents in the vicinity or farther away from that.
Yeah, I frankly don't know.
All right.
Yeah, I'm sorry about that.
Okay, good.
Yeah.
Nice project. Good luck with it.
Thank you.
All right, well, no, then we'll thank you for your questions and your presentations, and then we'll move on to the next speaker.
Yeah.
So I'd like to call Dr. Dilzar Kader to this front. Where are we again? Go there. And while I open his presentation, so Dr. Kader is from Iraq, and that's where he did his master and his PhD. And then he moved to Germany, and we were in touch. So finally, he received his Marie Curie grant. We'll tell you all about it, and he is working now in my group. So welcome, Dilzar.
Thanks to nice introducing me, and thanks for the committee that gave me the chance today to be here among you. It's a great pleasure for me to be here. In the next quarter hours, I would like to explain to you why I am here. Actually, I am here to have a good life, in another meaning, healthy, active life. Little about me. Here, I just need the pointer, I'm sorry. Oops, well, that's, okay. I am from north of Iraq, from Kurdistan region. And the pointer, I'm sorry, no, it's not working here. I am from Kurdistan region, I'm here. I'm from Kurdistan region, north of Iraq. I studied my master's degree in University of Mosul, north of Iraq, at 2003, and then I had my PhD degree, and as a dual site in between University of Zaho and University of Pavia in Italy. During my PhD at 2011, I traveled to Italy to do a part of my PhD in University of Pavia. After I finished that part, I went back again to finish my PhD in University of Zahwa 2013. After that, again, as a first postdoc, I did my first postdoc in Italy, in University of Pavia, with Professor Mocha. And after I finished, I went back again to Iraq. Then, after work, to work condition. Unfortunately, ISIS grew up in the region, and many people suffered, especially my people. And then, with my family, we decided to immigrate from our country to Europe. That's, I cannot say fortunately or unfortunately. Then I traveled with my family to south of Germany to live as a refugee. For more than 3 years as a refugee in Germany and searching for a job, finally, Professor Judith Sluimer, she could find me to have a chance for a collaboration. Then, after gathering and thinking about a project, we decided to apply for a grant and a project. Then, I moved to Maastricht to be here today among you. It's with a great pleasure, according to the collaboration. The collaboration built upon the idea and thinking about the project and giving rise an outcome of our project. And what can I add in my sight to her group that she missed with the group? In my expertise as a two-way knowledge exchange with my expertise in myography and contractility assessments and epifluorescence imaging techniques, and with her expertise in fibroblasts, we to rush into a main goal in vascular aging and a fibroblast in vascular dysfunction as a promoter. So, as you saw in the title, my project will be in between vascular aging and fibroblasts. How it happened? It happened during networking of scholarship at risk, connecting to Deutsche Research Council, NOW, for academic opportunities. I applied to Hestia Impulse for a refuge in science. And with the expertise of Judith's group, we applied to Marie Curie Individual Fellowship as a career restart panel. And I started at 15th of June of this year. Our project, the vascular aging and fibrosis generally comprise the most cardiovascular diseases. And the aging related to fibroblast plasticity and its role in fibrosis are generally unknown. And the vascular fibrosis generally characterized by reducing the lumen and considering with the matrix or the adventitial that producing fibroblast and collagen accumulation. And I don't know why it's happened. With lacking some informations, we want to fill in between the gaps relating to vascular aging induced fibroblast change with age. Interestingly, the deposition of matrix appeared in the home of fibroblast adventitia. And as a fibroblast, related changes in vascular aging generally unknown, in addition to their role to producing extracellular matrix. However, fibroblasts may promote some other functions, such as inflammation and angiogenesis. In the next few slides, I would like to highlight some pilot data and results that's already done by Judith Group, as we see here. Single-cell sequencing technique using starting from isolating the organs into trajectory analysis. In this figure, we see cell populations of smooth muscle cells and fibroblasts, which each cell equal to 1 dot. And the cells which are similar in expressing are profiles and grouped. Here in this image, we see the expression of marker. Here in the myosin heat chain 11 markers, which highly expressed in smooth muscle cells, and collagen 1 alpha 2 in fibroblasts. In this figure, we see the trajectory analysis of adventitial fibroblasts, representing 12 clusters in 3 populations. Regarding to the trajectory, we see that here in this figure that the percentage of fibroblasts are increased in different populations during the age, especially in population track 1, which generally correlates to vascular development. So on, it's important to assess age-inducing change in fibroblast subsets to establish a key hypothesis that the fibroblast function and plasticity are essential for blood homeostasis and changing with vascular aging. And here we put, I just put a key with a key stick referring to the key fibroblast bearing an information that may change in age and giving rise for more exploitation in future study. Focusing on our scientific objectives, we aimed to comprehensively study aging related to fibroblasts, plasticity, and their function in aging during using different techniques such as single-cell sequencing, fibroscreens, and contractility assessments, maybe different technique also, which we were going to be used to study in vivo, in vitro, and ex vivo, so on, exploiting unimaginable details. For the first aim, I don't know what's happening here, to purify the fibroblast populations and the functional fibroblasts in related to with aging comparing to young. I don't know why. For subsetting the function, the aim 2, we will do a culture of fibroblasts in young and aged mice, aortic part, and we will compare the functional fibroblasts in age and young, analyzing with fibroscreens. And for the third aim, in addition to in vivo study with pulse wave velocity, we will focus on a contractility assessment using myography in different cases with age and young carotid segments, which we will apply to wire myography in addition to compare the deficiency of population 1 marker dipeptidase 4, DPP4, and comparing in knocked out mice with DPP4 to an age and versus wild types. In addition, so we will go further to compare the segments, carotid segments, with adventitia in situ and without, to assess the role of fibroblasts, which mainly residing in fibroblasts as a home place. By that, it will give me a time to add to my skills that during the last months I add to my skill many techniques, and it will be a great chance to give me a chance for my recent and future career to be as professional as an independent researcher. By that, I would like to stand and thanks to whom helped me to get started and to be here today among you, and I want to thanks to whom helped me in the lab to teaching me and learning me as a group in new techniques and to adding my skills, and thanks for your attention.
Thanks, Deelzar, for the overview. There, of course, is still the opportunity to post questions in the chat. Our chat master is ready for you. But in the meantime, there may be some questions from the audience. Yes, Leon, please.
Very nice presentation. So how do you translate the in vitro work, so the fibroblast, specifically focusing on the fibroblast, with the myograph experiments? Because there you have a whole vessel, including extracellular matrix, including fibroblasts, but also other cell types like the endothelial cells, smooth muscle cells, and pericytes. So how do you compare these in vitro with ex vivo experiments, and would there not be a better setup to test fibroblast stiffness, so cellular stiffness, rather than vessel stiffness?
Thanks a lot. Yeah, as you saw, it's a bit huge project, and actually it will depend on our preliminary data, what we will have, but actually we have another plan, for example, to do it in vitro with cell culture isolating, for example, fibroblasts, comparing to endothelial cells and also smooth muscle cells, and also if we have enough time, or maybe in the future exploitation, to do it in situ, for example, with the segments, aortic segments or cardiac segments in situ. That means the adventitia are there. But also there are some problems with in situ studies in addition to the culture cell alone. But also the other main goal is to study the fibroblasts and adventitia in the place, without removing it, comparing to the removed one, in addition comparing those also in the knocked out mice in young and age, and also in both genders. I hope I answered.
Yes, we can talk more about that during the coffee.
Yeah, of course.
But indeed, Leon has a really nice machine where we can also measure the stress on the cell itself, in addition to what you were saying with removing the adventitia with and without, and having the knockouts for the fibroblasts. So that would be a great way to collaborate in the future. Any other questions? Yeah, Cengiz, there's one from you? From me. Oh, great.
Question from me, not from online. Thank you very much for the talk. I was just wondering, where does your hypothesis with this dominant role of fibroblasts sit with the notion of there being an adventitial somatic stem cell population? Is that synonymous with the fibroblasts? So are they the same? You know, when you talk about if we were to talk about an adventitial stem cell, is that the same as your fibroblast, or is this a different cell? And then where do you perceive this in your research question?
I think it will be a different style.
A different cellular population.
Well, yeah, because the fibrosis present in all three populations, so it will depend to move from place to vascular and or to other places. So I think, in my opinion, it will be a different.
Okay, thank you.
Thank you too.
Are there any other questions from online? No. Maybe one last one because we have some time. So what type of patient do you think we could help in the end with this insight?
Yeah, I think, in my opinion, it's with the pulse wave velocity patients.
So patients with high blood pressure and more pulse wave velocity. Is that what you?
I don't think so.
Okay.
Yeah, not yet, at least.
All right. Good. Thanks a lot.
You too.
Obviously, I didn't just find Deelzar. I'm not sure if it was completely clear, but he was really actively looking for a job and listed himself in an NWO database, the Hestia refugee grant. And then I was looking for people with expertise that was complementary. And then but unfortunately, he was a German immigrant, so he couldn't really apply for the NWO. But then we thought, well, we should really do this, and we wrote this Marie Curie grant together also with with Elke, with the three of us. Elke Mars, my former PhD student. So thanks very much, and welcome to the team, and welcome to CARIM. And hope you can present your data in the posters in the next session. And of course, the same applies to all the other candidates. And I think we are having a little bit longer break, and we will start again at 3:45 with the program. Thanks a lot, everybody.
Thanks.
Announce this session, which is alternative research funding from HFL, Heart Foundation Limburg, and SWOL. We have 2 awardees, winners, and these winners are there for the grants, that's me, crowdfunding, and for the Harry Krijns Research Grant. You know, if you retire, then you come to Heaven's Gate, and then Peter, she says, oh, you were a good boy. And then they name a grant after you. And then on second thought, Peter, she said, oh, you were a very good boy. We give you 2 awards to be named after. And that's why there's now this Krijns crowdfunding awardee, Rogier Veldrop. He's going to present From Patient to Research, A Life with Laminopathy. This crowdfunding award will continue after, and Tilman already mentioned that Shahinda Hossein is this year's winner. The point we want to make is that if you want to receive this award, this crowdfunding award, then you have to come up with a very good idea, which the committee likes very much, and then you have to collect the money, and once your crowdfunding reaches over 25,000, then CARIM will spend another 25, so you grow to 50 at once. So come up with a good idea, go for crowdfunding, and then knock on the door with CARIM. Be then the first in the next year. We're talking about 2022, and then your crowdfunding will be doubled, or at least heightened with 25,000. Having said that, and I already announced you, Rogier, please take the floor and speak about your crowdfunding topic. Go ahead.
Okay, thank you very much, Harry, for this introduction, and I'm very glad that you haven't seen Heaven's Gate yet, so that's a good thing. Talking of such, I have, so that's, but I'm going to talk about this in this brief overview of a life with laminopathy. So I just want to point out my background in a nutshell. I started out with my academic career for a 4-year clinical chemistry education in Eindhoven, a 4-year molecular biology in Utrecht, at Utrecht University, and I commenced my PhD in coronavirus research, which was not a big thing at all back then. Still, I had to stop my PhD in Philadelphia due to heart failure, and that's why these buttons are there. After my heart failure, I came back and worked in Utrecht and in Eindhoven. I recommenced another study, another master in biomedical sciences, cardiovascular biology, here in Maastricht. And after that, I started a CARIM project, which is the cardiorenal syndrome analysis. And I hope to graduate within the next few months at a double degree from Aachen and Maastricht University. The title is A Life with Laminopathy, but what is a laminopathy? So I just want you to give a brief overview of a laminopathy. So here in blue, you would see a large blue nucleus, and the nucleus of the cell. And just near the nucleus, there is a mesh, the nuclear lamina. Nuclear lamina is very important because that transduces all forces applied to the cell. And so there is a chromatin anchor connection, but also connection to other parts of the mechanotransduction apparatus of a cell. And on the right side, you actually see what it looks like at the top right corner. So it's actually a dense network over there. When I would stain it from cells, and these are actually my own cells and own nuclei, I stained the YOL2, YOL2, that's your lemon network, and in green you see that, but these are not ordinary nuclei. You would see a lot of malformation, so you see a complete hole in the nucleus, but you also see extra blabbing, and you see little holes here in the nucleus, and so there are different types and different hallmarks of a laminopathy. This causes many problems, and that starts with the instability of the nucleus. And to what types of diseases this would lead is many different types of laminopathies, but the different types of laminopathies does not seem related. You have many different types of laminopathies, which due to time reasons I will not go into deeply, but you see a lot of different phenotypes over here. And probably at the right side, Progeria is probably the most well-known. So this is Sam Burns. Sam Burns is Progeria patient, which is also a laminopathy. And Sam Burns reached the age of 17. So he's just turned 17 at this picture. So it's a rapidly progressive aging disorder. But the most common problem with laminopathies and the most severe problems arise then with the cardiac phenotype. And over here you would see an LVAD, a left ventricular assist device. And on the left, that's actually me in the hospital because I had the laminopathy myself. And this is a left ventricular assist device, and on the small part it will be connected to the left ventricle. Over there, just where the sun is pretty much. So over here, and there is a impeller, and it will push the blood towards the aorta. And this is a machinery, so the machinery needs some batteries, and the batteries you see on the side here. So I had to live with batteries because the pump is electrically driven. So if you have an assist device, you just cannot go out for swimming. You cannot shower. You cannot walk in the rain. So those are difficult situations what you have to live with the left ventricular assist device. And that led to more problems. So also the right side of my heart started failing. When the right side also fails, there is also an RVAD, right ventricular assist device. But the chances for me to survive that surgery was already really, really limited with a prognosis of a few weeks. So we decided not to do that and put myself on the highest urgency for heart transplant. And eventually I got a heart transplant, and with a collaboration with Utrecht, I got pictures and received slices of my own heart. This is my own heart. And you see an extreme left ventricular dilatation, but also the right side is usually dilated. And on this side here, you would see the entrance of the LVAD as well. So I got a heart transplant, and so from a PhD in the United States towards a heart failure patient, leading to a heart transplant, that was a huge setback. So for me, this was a moment where you just realize, okay, first you strive towards the highest, and you want to have an academic career. You want to be successful in every way you can think of, and in the end, you're just fighting for your life. Not for weeks, but for many months. And I've been in the hospital for over a year before I received my donor heart. So this was a very difficult moment. And just afterwards, this was what it looked like. So I was 52 kilograms while being over 190 in height. So this was a very tough time. And the only thing I wanted is to get back into life, start living again. Because this period of time took 8 years. So this was a very tough period of time, but it makes you think, what do I really want? And for me, that was going back to academia. So I wanted to go back to academia, and when you are at academia, of course, everybody recognizes this part. You want to have funding for your research, because you think you have really nice funding, really nice research, but of course it costs a lot of money. And unfortunately, or fortunately, research is very expensive. So we all know the drill, we all know the problems in the research, but of course you want to have a lot of funding for to generate attention, less waste and better future and better machines for better research and better output. But it's very important to realize that always, regardless the money, the best idea in your head, you really want to pursue, and you want to make your research meaningful. So I went from coronavirus into the cardiovascular biology, and then I started investigating my own disease. I don't investigate only and solely my own disease, but I started focusing on the mechanobiology of mononuclear or monogenetic cardiac diseases, of which laminopathy is one. So there are a lot of funding opportunities, and I think we should be very, very happy. I've seen many universities in the past, Eindhoven, Utrecht, Nijmegen, Philadelphia, but at CARIM, that's the only institute so far where we actually get a newsletter which lists all funding opportunities. So I think we should make use of that a lot. And of course, the NWO, Dutch Heart Foundation, European grants, and those are already mentioned. That's a really nice funding strategy as well at the EU. There are also registered funds and company funds, and those are less known, but it's very interesting to take a look at those as well. But you can also start your own crowdfunding, as I did. So first I went to University Fonds Limburg, Zwol, and I started up a web page. I started up making my story known, and I wanted to proceed and see if I could make 10,000 euros. That was the primary aim. Then I wanted to see if we could get like 25, and that was in the beginning that was tough because you wanted to have a small impact on family and friends, but pretty much half of my family already died due to this disease. So the amount of money by family was, of course, very limited. So we wanted to start exposure. So we went to Brightlands, and we got an interview. And from there, one thing led to another. And there was a television show that had a charity program. And for the charity program, we wanted to see if we could compete. And I was, in the end, I was in the top 10, but only the first 3 could compete for actual funding. So that was already okay, but it was nice to be exposed at that level, but unfortunately no funding there. And that changed when I was at the late night program Op 1, because at that moment, during the evening, the crowdfunding really started to explode. So from a few thousand euros to 15,000 to 20,000, all in pretty much 15 minutes on national television. So that was very important. So during the television show, or I thought a few hours later, we actually got the 25,000 euros. So that was a huge moment because indeed, as mentioned before, oh, DG left there, the CARIM crowdfunding doubler, I was very happy to receive that. So from 25K, we went to 50K immediately. So there was a huge stronghold over there. So then it's getting impactful, and that was really, really nice. Because you've been on television and because you go further with a lot of interviews, a charity cycler actually helped me out, and he started a race of 1,000 kilometers. And 1,000 kilometers, he knows a lot of people. They all invested a little bit of money, and because he knows so many people in this field, and he was so enthusiastic. So in the end, we got a 10,000 euro check in the end, which we celebrated in front of our university, completely COVID-proof. In the end, I think I could have made 5 or 6 of those slides. Did a lot of exposure, a lot of different interviews and television shows. And that, in the end, it led to a check to our by now former president of over 80,000 euros. And so there was a great end of that crowdfunding campaign. But what do we actually want to do with the money? And I'm just going to explain it really briefly. If we have a patient or could even be an asymptomatic, because it's horrible when you call somebody a patient while it's a carrier of a genetic mutation. But it can also be asymptomatic. So we just have a tube of blood. From the tube of blood, we want to, you have the point here as well. So we want to, tube of blood, we isolate PBMCs, and from the PBMCs, we introduce certain factors that turn back the time of the genetic components of the cell. So we reprogram the cells into iPSCs. From iPSCs, you can derive and differentiate the cells into pretty much every cell type you want. And in my interest, it's the cardiomyocyte. From the cardiomyocytes, of course, we want to bring it back to the patient, and that could be as a therapeutic plaster, that could be in many different ways of introduction, but that's where we are now at the moment, to really give it back to the patient and try to optimize a cell therapy for the future. But also the supernatant out of the cardiomyocytes is very important. And for example, the exosomes, the smallest extracellular vesicles, are really interesting to take a look at to have a cell-free therapy. And in the end, when you have the cardiomyocytes, you actually see this under the microscope, which is, of course, especially the first times, it is an amazing view to have these beating cardiomyocytes ready to go, which is patient-specific. So these are actually my own cardiomyocytes, and then in young phenotype. And then just a brief 2 steps of research. So the first one, you just saw a Z-stack of a cardiomyocyte bundle, and that worked out nicely. And here you see again the strange nuclei of the cells. So these were again with the laminopathy. And you can make these images out of your beating cardiomyocytes. So in the end, you want to investigate, you want to do research. There's a lot of time and effort, of course, in each research, but you also want to invest time in your crowdfunding. So what to do and what will be important, and is there time enough, and is it fun enough to actually pursue a proper crowdfunding? And that is, for example, joined HFL, and HFL had loop met je dokter, work with your physician. So that was actually fun to do. That was a few months ago, and by now I had an own page, which is hartonderzoek.nl, and that's then you will be redirected to the Hart- en Vatenonderzoekfonds Limburg, so the Health Foundation Limburg, and I already started a nice crowdfunding campaign over there as well. So there's a lot of initiatives and a lot of fun things to do over there. So what to take home? If you actually want to start a proper crowdfunding, take the action, and there's a lot of initiatives and stimuli, as we just mentioned, with Harry Krijns and Tilman Harking. But you have to take action, you have to do it yourself. So, and it's really time investing, but it's actually very, very rewarding. You need to have a proper plan, and I'm always open for discussion if you have a proper plan how to fine-tune and shape it. It's a matter of exposing yourself. So in the beginning, the pictures you've seen, of course, that was hard for me as well in the beginning. These pictures weren't in there in the beginning, but you need to be sure how to bring the message across. To know the audience and where are your potential crowdfunding donors. So that's very good to have that insightful. And you have to create a network and be interested in the network as well. So if patients that come to our department for blood draw, actually talk to them. What do they feel? If I have students that I supervise, I bring them over to the patients, have interaction. We are not just solely looking at cells in our lab, you're looking at patient material. And I think the interaction to actually know that you're working with patient material is very important. Show them the results as well. Try to bridge academia and patients. I think that's very important. And keep in touch with them, not just a blood draw and bye and won't see you again. No, those people want to be involved. That's why they give the blood, not because of your research. They want a solution in the end, and they want to keep updated. So I think that's very important. There are many ways to keep in touch with your patients that you or treat as a cardiologist, for example, but also as a researcher. There is a lot of opportunities. So I started my own foundation a few months ago. This is lmnacardiac.org. And there we have a lot of different LMNA patients, which is considered still a rare disease. And a rare disease, we have already 250 patients within the last few weeks. So these people are actually seeking information because it is a rare disease, so they want to be informed. So I think that's very good to keep in touch with your audience. So for now, I want to thank Leon Scheurers, of course, and the whole group in supporting me, and Tilman Hekking and Harry Kreins, of course, also for the doubler in there. So at SWOL, Guido van der Broek, Milou Schreuders, and Bojan Jansen, and of course of Health Foundation Limburg, Elle van der Ven, and Meke Koman, and of course all the people that donated, not only money, but also my heart. Thank you.
Thank you very much for this personal story, the cutting-edge research you're doing, and all the examples you gave on how to raise money. Really impressed. You already got your 25,000, is it? Yeah. Okay, so I'm not going to. I'm not going to give them now. So but thank you very much for excellent presentation and a good example. And one point you have to also take from the presentation, get on national TV, because that's also how Sheinda got her got her crowdfunding going. So if you have this good idea, get on national TV and then knock on the door at CARIM. Thank you very much again, Rogier. Now, the Harry Kreins research grant. This is a grant which is then provided by the Health Foundation Limburg, which is a foundation here in this region, connected to the MUMC, connected to the research which is happening there. Health Foundation Limburg wants to support researchers, amongst others in the cardiovascular field, and the foundation, they put in place this research grant, which was named after me. And I have to say it's sponsored by Bayer, Amgen, and Sanofi. We had at least 10 excellent applications. You must know this is a research grant which helps beginning researchers with a small, well, a big amount of money, it's 25,000, amount of money to turn the first pages of their research, to start researching under the supervision of an established researcher. We had 10 applications. The committee had a big problem choosing the very best because we had a lot of good applications, but we choose for Sophie van der Walle. And she is here in the audience, she is in the first row, and she is she's going to tell you her story, her first page turns of research, and you will understand why we choose her. Okay, Sophie, go ahead.
Hello everyone, I'm Sophie van der Walle, a fifth year medical student at the University of Maastricht. And may I start with the fact that I think it's an honor to be standing right here today and tell you something about this amazing project, the winner of the Harry Kreins Research Grant of 2021. So thank you for that, and also thank you to the Health Foundation Limburg. The landmark study is a study that I'm very, very proud of, because it may have a huge impact on the present-day healthcare in the future. And now I want to take you directly to the practical problem. Above you see 3 pictures representing the aortic dilation. And on the first picture you see a CT scan, which shows the aortic diameter. However, we come across several problems, as for example, low temporal resolution, a low frame rate, and we also have to deal with a spatial picture. However, the organ is very dynamic. And on the second picture, we see a 4D flow MRI, which is a very nice scan, which represents, for example, wall shear stress or the pressures within the aorta. However, we come across several problems as well. For example, low spatial resolution, which results in loss of anatomical information and specific details. We cannot measure the axial strain, and we cannot measure the increase in length or in other directions. And we cannot measure the elasticity as well. And on the third picture, we see an ultrasound. But we also come across several problems over here. For example, a low temporal resolution, a low spatial resolution, loss of anatomical information, etc., etc. Below, we see 3 pictures which represent right atrial dilation, which is an important factor for the manifestation of atrial fibrillation. And again, we come across the same problems: a low temporal resolution, a low spatial resolution, loss of anatomical information, etc., etc. And we cannot see the different advantages of the different imaging techniques coming together in one picture. Well, we can say that cardiovascular diseases are now being diagnosed in end stages. And this is caused by the fact that we diagnose on behalf of anatomical information, anatomical parameters. So it's time to change that, because this takes us to difficult positions, for example, aortic dissection, a very dangerous disease with a mortality of almost 70%. When we take a look within this population, we see that more than 90% of the people in acute situations still do not fulfill the criteria for preventive aortic surgery. So we cannot provide people with the care that they need. And when we take a look at atrial fibrillation, we see that we diagnose people when they present themselves with palpitations, or in 30% of the cases, when they present themselves with stroke, for example, which is a complication caused by atrial fibrillation. So we have to do something about this. Well, we and our currently used imaging techniques are just very limited in possibilities. And this is due to the fact that we have to deal with a very complex, geometrical, 3-dimensional shaped organ with irregularities. We also have to deal with the environment, which is very dynamic. What we do know is that we now have to focus on the patient-specific dynamic parameters in order to create more knowledge in these specific diseases. Earlier research within the Maastricht University Medical Center showed us that aortic elongation is a much better parameter for the prediction of aortic dissection instead of aortic diameter. When we take a look at atrial fibrillation, we see that not only atrial dilation, but also strain rate, ejection fraction, and identification of proarrhythmic substrate are very important parameters. So I think we should focus on these parameters instead. What we should do, what we try to do, is to augmentate the different imaging techniques that we're using nowadays in order to quantify the local tissue changes and correlate these to the stimuli that provide and cause these tissue changes. We also try to make the different advantages of the different imaging techniques correlate in order to identify patients at risk of a certain condition and treat preventively. Well, how are we going to do that then? We've already seen that different materials that we use during open heart surgery can easily be seen on MRI and CT scans. When we take these materials and put them systematically in patients, we can use them as a sort of markers. Well, we will first do that in the right atrium with 10 markers, and at the aorta ascendens with 20 markers. because these are the most easily accessible and therefore also the most safe to assess. Well, this provides the ability to take several measurements between these markers. We can follow them in place and in time per person. And these measurements can be taken in all directions, as you can see in the right picture. Well, with this knowledge, we can then quantify and detect these local tissue changes and measure them per reduced anatomical segment, which makes possible to do 21 new measurements in the right atrium and 65 new measurements in the ascending aorta. So that's a lot. Why is this so important then? Well, until the 20th century, we thought that genetics was everything. We thought that genetics would cause our whole phenotype. However, we got rid of this idea quite a long time ago. And we now know that external stimuli are at least as important. That's exactly the reason why we tend to look at, for example, lifestyle, education, and comorbidity, as for example, hypertension, wall shear stress, 4D flow, geometrical changes, etc., etc. What we try to do is create a kind of algorithm with all these comorbidities and external stimuli, in which we can identify people at pre-stages of their phenotype, at pre-stages as, for example, aortic aneurysm, aortic dissection, or atrial fibrillation. I will have the role as a coordinator, which means that I'm responsible for the medical ethical approval, for the creation and management of a database, for the planning of interdisciplinary meetings, etc., etc. And Dr. Bidar will be my supervisor. As I already told you, I'm still a medical student, so I'm currently doing my clinical rotations. And I've now almost finished all my general clinical rotations, which means that I've only got left my 2 clinical rotations of choice, my scientific internship and my final internship, which I will all use for this project. I've been involved in several projects for the last few years in cardiology, in cardiothoracic surgery, and I think it's an honor to finally have my own project with such a huge impact. And I will use this project therefore as my PhD, and I will continue this after I finish my studies. There are several corporations within the Maastricht University Medical Center which we can use for our project as well, as for example the cooperation between biomedical engineering, cardiothoracic surgery, biochemistry, and radiology, which has led to a biobank for patients with aortic aneurysm. And there's also a close collaboration between electrophysiology, cardiology, cardiothoracic surgery, which has led to, for example, the FibriCheck, the rhythm monitoring, and the identification of a proarrhythmic substrate for atrial fibrillation, which we can also use in our project. Well, we try to have medical ethical approval before May 2022. And we will do the inclusion between May 2022 and 2023. And this will consist of 40 patients, 20 coronary artery bypass grafts, and 20 aorta valve replacements. They will receive the general surgery, and in addition to the general surgery, they will receive placement of additional landmarks. And also tissue samples and blood samples will be taken. During the hospital stay, they will receive and undergo MRI, CT scan, and ultrasound. And we will start with the rhythm monitoring. 1 year postoperative, they will again receive CT and ultrasound, and 2 year postoperative, they will again receive MRI, CT, and ultrasound, and the rhythm monitoring will be continued all along. It's good to know that the ultrasound is already part of the general care that people receive after their cardiac surgery, so no extra costs are needed for this part of our study. The money that we received from the Harry Kreins grant will be used to finance 2 MRI scans and 3 CT scans per person. So thank you very much for that. Now that I've given you an overview of the complete study, I want to focus on the point and the main goal of our study. We want to identify patients at risk of a certain condition, which means that we make a shift from curative care to preventive treatment. And all this preventive treatment altogether will make a decrease in acute situations and therefore will provide as much as planned care as possible to the local community, which will finally make the mortality rates drop. Thank you.
Well, thank you very much, Sophie. Is there a question from the floor? Then you have to take the mic, Frits. Professor Prinsen.
Hi, thank you very much. Great presentation, interesting topic. I wonder, I'm interested in working with deformation of the heart for many years. You mentioned markers to put on the left, on the atrium and on the aorta. What kind of markers are they?
There are things that we already use during the open heart surgery, so they are safe. And it will be.
What kind of material?
They're kind of clips, like a material clip, and a kind of stitches. And they can easily be seen on MRI and CT scans.
And you can see them, but then you want to, if I understand properly, you want to follow them in time.
Mm-hmm.
And how are you going to measure that? What kind of software or so do you use to get this three-dimensional anatomy and motion maybe?
Yeah, so the markers are kind of a landmark, and this provides the possibility to put the different scans, as for example CT scan and MRI, on top of each other. And because of this possibility, it will be possible to take measurements between these indeed.
How do you do that?
I don't really know what kind of program we will use for these measurements. We still have to check that. We have to find out what will work the best. So I can come back to that later.
Okay, well, at least please know that there is experience in physiology and biomedical engineering. For example, this X-ray marker, like we actually used at that time, golden bullets, small golden bullets at multiple sites to follow them. So there is experience in software which may facilitate your analysis.
Okay, thank you very much.
You have your first new collaborator on the project.
Yeah.
That's Professor Prinsen. Thank you very much. There's one more question from the chat.
Indeed, there's a question from the internet, which comes from Shiv Parikh, who says, very nice presentation. He has a question regarding using clips on the tissues. What are the additional advantages of clips over regular medical images for characterizing tissue?
So by using the clips, you are able to place the different scans on top of each other. Otherwise, that would not be possible. And in addition to that, because of these clips, you can reduce the anatomical segments where you take measurements in between. So that was not possible in the future, and we could only take measurements in like very large anatomical segments. And now we can reduce these segments, so the final quantification becomes much more detailed.
I'm sure he thanks you for the question, but it's not quite on the chat yet. But thank you. Yeah.
Yes, thank you very much. Now we come 5. Oh yeah, there's a break. But before we go to the break, I want to present to Sophie the award. And that's, maybe you step forward.
Thank you very much.
Thank you very much. And it's a 5 minutes break, so in 4 minutes time we come back here. So better stay put and wait for the next presentation.
I'm happy that you accepted our invitation and found time to participate in this CARIM symposium. So Barbara is a British Heart Foundation professor to John Radcliffe Hospital in Oxford. She's also the past president of the European Society of Cardiology. She graduated in medicine at the University of Pavia in Italy, and afterwards she moved to Oxford for further clinical and research training. And what initially was planned to be a short stay has developed in a PhD degree in cardiovascular medicine at the Pembroke College at the University of Oxford, and afterwards in a beautiful and impressive lifetime career in the UK. As an outstanding leader in the field of cardiovascular medicine, Barbara has been invited to give many keynote lectures and received many prestigious awards. She's a passionate supporter of the career of women in science, both locally and within the European Society for Cardiology, also placing a lot of effort on establishing initiatives aimed at supporting the career of young clinicians and scientists across Europe. Besides being intensely involved in many activities, she's also founder and core organizer of the Frontiers in Cardiovascular Biology meetings, the FCBB. So I could take the next hour to tell you about all the important achievements in Barbara's career, but I think we can better give her the floor so that she can share with us her passion for and her radical views on atrial fibrillation. Barbara, please, thank you so much for being here, and the floor is yours.
Thank you. Thank you very much indeed, and congratulations again on your professorship.
Thank you.
Here are my slides. So I can hear a huge echo. I don't know whether you have the same problem. Do you?
Also, please turn off the handheld microphone.
Shall I start again? This is much better.
Yes, go ahead.
Thank you very much. Yeah, thank you so much. And so thank you for the honor of giving this prestigious lecture and the association for a very eminent physiologist, vascular physiologist, I would say, and also for all the help and fruitful collaborations that I have had with physiologists and cardiologists in Maastricht over the year. I mean, it has been one of the, definitely one of the highlights of my career. So, Radical Views on Atrial Fibrillation is a title that has a double edge, if you like. On the one hand, it reflects my interest in free radical biologist in the myocardium, and on the other also an interest, an attitude, if you like, that I know is very much shared by electrophysiologists in Maastricht, to look a little bit at the lateral side of atrial fibrillation, if you like, and to be not quite so conventional in the outlook. Of course, we are all children of our time, and we all have some views that come from the knowledge that has been put forward before us, but equally the way we interpret that knowledge can change. So, every talk on atrial fibrillation starts with it's a very common arrhythmia. And it's true, the prevalence is very high and increasing with the aging of the population and the increased prevalence of its risk factors, and particularly now obesity. There are things that are associated, morbidity that are associated with atrial fibrillation, and in some cases we think causally. And one is indeed stroke, that we think we understand, and heart failure as well. Why is atrial fibrillation associated with an increased risk of myocardial infarction is less clear. But overall, it is not a benign arrhythmia. It is associated with a worse prognosis altogether. And, years of trials and tribulations, and we're still, somewhat stuck with anticoagulation as the only treatment that has been consistently shown to have a favorable impact on the prognosis of IF patients. And so, every time I look at this, I think we haven't done well, because at the time where we have seen amongst the steepest reduction in cardiovascular death in Europe and elsewhere, as indicated here, the global burden of disease has indicated an increase in the age-adjusted mortality for atrial fibrillation, not only in the developing countries where the statistics are, a little bit not quite so strong or robust, but in the developed countries as well, both for males and females. So what have we done wrong and how can we do better? And like many, I have started this journey with Maastricht in mind, and with the atrial fibrillation begets atrial fibrillation story. And you know better than I do that once the atrial fibrillation starts and becomes persistent, it does induce changes, electrical and structural, in the atrial myocardium that maintains the arrhythmia essentially. And in a nutshell, together with fibrosis from the electrical side, these have been really summarized in, we have a shorter action potential duration and refractory period, and we have a slower conduction velocity, and that will favor re-entry, and that is that. Right. So despite all of this, though, and the fact that this remodeling has been described in many species, including humans, and in many models of atrial fibrillation, what it is actually triggering it has really escaped us. And if we think that there may be an advantage in preventing it, then we need to know what is the cause and ideally go at the very root rather than downstream. And so we undertook a number of studies, and we started with a hypothesis. One has to start somewhere, and this was befitting our expertise. And the fact that there were some preliminary studies that indicated that indeed there was an increase in the myocardial formation of reactive oxygen species, as well as a reduction in nitric oxide production in the atrial myocardium of animal models with atrial fibrillation. So the starting hypothesis was altered myocardial nitroso-redox states may contribute to the myocardial substrate that subtends atrial fibrillation-induced electrical remodeling. And many years ago, I came to Maastricht, where I gave a grand round, really focused on our work on reactive oxygen species. And today I would focus on nitric oxide. So we know that nitric oxide and reactive oxygen species are involved in many of the mechanisms that are also involved in atrial fibrillation-induced remodeling, and particularly electrical characteristic of the myocardium, energetics, and also fibrosis. And the way we approach the problem is usually well, we do stick our neck out we take risks. We take risks by putting together quite a big structure that requires significant investment, both in time, emotion, and money, in order to answer the question. Typically, we start from human atrial tissue that we obtain from patients that undergo cardiac surgery. And from that, we will isolate cells, do molecular studies, small-scale modeling, and patch clamping. And if something looks promising, we would then look at a mouse model where we use to answer the question, is this mechanism sufficient to induce or favor the induction of atrial fibrillation? And all of this, we need to thank Sander Verheule, who has been instrumental in telling us essentially how to do it. And then if we're lucky, we Uli would then agree to share some tissue from an animal model of goat model of atrial fibrillation when available. And that has also been very important because the mouse has, very large limitations. And then we go back to humans, either by doing deep phenotyping with state-of-the-art magnetic resonance imaging and CT, but also clinical trials and more recently Mendelian randomizations in the context of UK Biobank, again, as an alternative to the mouse to establish causality. So from that, what have we found? Well, looking at tissue from goats with atrial fibrillation and controls in sinus rhythm and atrial fibrillation for 2 weeks and 6 months, what we found is that both in the right atrium and in the left atrium, there is a reduction in nitric oxide synthesis, as indicated here, that is sustained over time. This is different from reactive oxygen species from some sources that really are only increased in the early stages. Then we went to our human tissue bank, and what we found was pretty much the same, and that is that in the right atrial appendage of patients with atrial fibrillation, the synthesis of nitric oxide was significantly reduced. If we then look at the sources of nitric oxide production in the myocardium, and we have two constitutive enzymes producing nitric oxide, the so-called endothelial isoform and the so-called neuronal isoforms, we see that the protein content of the endothelial isoform is unchanged, whereas the protein content of the neuronal is almost zeroed. And so that was unexpected and made us think that it might be something related to the parasympathetic ganglia that also have a large proportion of NOS in those nerve terminals. But actually, when we isolated atrial myocytes from patients in sinus rhythm or persistent long-standing atrial fibrillation, then we saw that it really is in the myocytes that it is reduced, as indicated here. So, there is, from here, we know, okay, loss of myocardial NOS must be the reason why there is a reduction in atrial nitric oxide synthesis both in human and in other animal models of atrial fibrillation. And the question is, what do we know about NOS? Well, NOS, we have been working on NOS in the myocardium and vasculature since the beginning of this century, which sounds a long time indeed. And what we have discovered is that the neuronal isoform is can be found both close to the sarcoplasmic reticulum in the proximity of the ryanodine receptors and also in the sarcolemma, particularly in atrial myocytes, with a number of functions which even surprised me, because it keeps coming back with doing something else, and is always this isoform rather than the endothelial isoforms that is the culprit. So we have found that it is involved in the regulation of excitation contraction coupling. It has an effect on the calcium current, on the ryanodine receptors. It has an effect on post-myocardial left ventricular remodeling, on phospholamban and phosphorylation. It has an effect on flow-independent peripheral and coronary resistance, for instance. And recently, we've also found that it has an effect on myocardial energetic and glucose uptake through the GLUT1 receptors in diabetic hearts. So on top of that, it also has an effect on potassium current in the atrial myocardium. But what is it that would make nNOS disappear in the myocardium? And again, going back to what we already know, we have to say, well, what do we know about nNOS protein stability and what controls it? And we know from patients with Duchenne muscular dystrophy that nNOS is tethered to the membrane via the dystrophin-syntrophin complex, and that if we lose dystrophin, we would then also lose nNOS. Or so we thought, because that is another longer story, but unpublished yet. So we looked, therefore, at dystrophin, thinking, could it be that the dystrophin is reduced in the human fibrillating myocardium? And indeed it is, as it is shown here. There is about a 60% reduction in dystrophin. And you say, does it matter? Well, it seems that it matters not directly, but indirectly via its effect on nNOS and the loss of sarcolemma localization of nNOS under these circumstances. So you can see here that if you inhibit nNOS with S-methylthioacetylcholine, you have a reduction in action potential duration in myocytes from patients in sinus rhythm, but not in atrial fibrillation. And that if you take a mouse with an nNOS gene deletion, it would have a higher inducibility of atrial fibrillation in the absence of structural remodeling. So what happens to the ion currents? And so it actually has an effect on several potassium and currents and on the calcium current. You can see here that inhibition on neuronal nitric oxide synthase increases AK1, AKUR, to some extent also ITO and the calcium current. And all of this obviously it's difficult to know what it is actually causing the reduction in action potential duration, because the effects could be contrasting. And so we resorted to our collaboration with Blanca Rodriguez in Oxford for modeling. And what the modeling has shown is that it is mostly the effect of nNOS-derived NO on IKUR and IK1 that reproduces the change in both the action potential duration of 50% and 90% repolarization that we see in experimental data. So kind of interesting in a way, and then again, what is it that is causing the reduction in dystrophin? And we could go on, but I go straight to the punchline, which is that at the end, we found that in the presence of atrial fibrillation, there is an upregulation of microRNA 31 it's also both in myocytes and fibroblasts. And that in the myocytes, this microRNA induces translational repression of dystrophin. This is something that had already been discovered in the field of Duchenne muscular dystrophy. But also, I mean, it also has a direct effect on nNOS. So the reduction in dystrophin would cause a change of location, if you like, you know, of nNOS that goes into the cytoplasm, is ubiquitinated and degraded by the proteasome. But then the link of the microRNA 31 to the 5' UTR region of nNOS accelerates the messenger's decay. So these two factors together cause a massive reduction in the nitric oxide production in the fibrillating myocardium that then has an impact and contributes to electrical remodeling through the process that I have described. And as I said, this came, Svetlana Riley and Xin Liu were the main author of this paper, which was done in collaboration with Maastricht. But why am I talking about something that has already been published? Because there is a but. And the but is related to the fact that in that paper, we really focused exclusively, at least on the human side, in right atrial myocytes. And so the question is, would the same occur in the left atrial myocytes? And we thought, well, of course, we will check, but, you know, for sure, that is, you know, the nitric oxide decreases on both sides, and so the effect must be the same. And in fact, there is nothing like doing an experiment, because when you do the experiment, you find that that is absolutely not the case. So the reduction in action potential duration is only pertinent, as you can see here, to the right atrial myocytes. On the left, nNOS inhibition does not cause any change in action potential duration. So as a result, we have a loss of the gradient in action potential duration between the right atrium, where normally have a longer action potential duration, and the left atrium, which seems to be dependent on the fact that the nNOS activity is higher on the right in humans, and the AKUR, which seems to be mostly responsible, maybe not entirely, but mostly responsible for the effect of nNOS on action potential duration, is higher on the right than on the left. So again, resorting to modeling to find out what does it actually mean. You know, what does it mean for atrial fibrillation to lose the gradient or to have a reduction in the gradient in action potential duration between the atria? And the modeling suggests that this favors the maintenance of atrial fibrillation, which also has a higher dominant frequency and a longer duration. Note that the duration in this simulation is capped to 3 seconds, so the differences are actually underestimated. And then, you know, if you then take all of this and you look at also what happens, you find that there may also be directly or indirectly effects on the sodium current, because, you know, there is a significant difference in the right only in resting membrane potential, which is then associated with a faster rate of rise of the action potential in the presence of nNOS inhibition, largest overshoot, and a larger action potential amplitude. So all of this, you know, again, looking at modeling, would be expected somehow to induce a reduction, or to be associated with a reduction in the duration of the P wave. And, you know, that was somewhat troubling because we thought, well, generally we have in atrial fibrillation, the concept that we have in our mind is that there is a slower conduction velocity, a longer P wave. But actually, there are papers out there suggesting that there is both. Both a short P wave duration and a long P wave duration and PR intervals can be associated with an increased risk of incident IF or recurrent IF after pulmonary vein isolation, as indicated by this paper. And you can see here another Maastricht usual suspects is a co-author. So again, the paper refers to epidemiological studies from the Copenhagen cohort that indicates that in patients referred to hospital for an ECG, the relationship between the P wave duration and the PR interval and atrial fibrillation is of U shape or a V shape, if you like. So that both shorter P wave duration and PR interval and longer are associated with an increased risk of atrial fibrillation. But of course, under these circumstances, one never knows what is the chicken and what is the egg. In other words, do we have here comorbidity, say heart failure, that would prolong PR interval and P wave duration and be associated with atrial fibrillation? So is there a confounding factor or is it a direct effect of the interval, and so the electrical makeup, if you like, that is causing this? And equally, there could be reverse causality, because in these epidemiological studies, patients with atrial fibrillation or a history of atrial fibrillation treated for atrial fibrillation were not excluded. So the only way to find out really is via a genetic study, and that is because genetics comes first. And so one has problems of pleiotropy and other confounders, but not quite to the same extent, and they are more controllable. So for that, therefore, we moved from this experimental loop of the circle to really work on large databases in UK Biobank. And this was actually very good because it coincided with the pandemic and the closure of the labs in the UK. And it was very good that we could be quite so flexible to move operations on big data and therefore don't waste time. So particularly good for students, PhD students, where time is particularly precious. So we turned to UK Biobank, that you would know is an open resource. Everybody can access this data by just paying a small handling fee for the data, and includes a very rich resource of data from 500,000 men and women that were recruited and assessed the first time between 2016 and 2010, where they were between 40 and 69 years old. And we know a lot about these participants, anything from the medical education history to the medication to the cognition, and we have what they're called enhancements, so that all 500,000 had baseline biochemistry biomarkers measured. 500,000 are genotyped, 200,000. The whole genome sequencing of 200,000 has just been released. We have the genotyping for 500,000, and we also have whole exome sequencing. 100,000 of them have MRI of the brain, heart, and body. And in this cohort, we have been funded to do a long-term ECG monitoring to look at the relationship between silent arrhythmias and cognition and cognitive decline, as well as brain imaging. But this will be coming in the next 2 years, will be completed. So we looked at, in this case, the atrial fibrillation. We know who has atrial fibrillation, at least clinically, in this group. And we ask ourselves the question, are genetically determined differences in the ECG P wave and PR interval linked to atrial fibrillation, and in which direction? And then we did a number of other studies making sure that this connection was not through changes in atrial structure or other risk factors for atrial fibrillation. So essentially, we looked at the genetic score that was available in the literature, all the GWAS that have been done describing the genetic influence on the QT interval, the P wave duration, and the PR interval. We then validated these scores in now a cohort, the UK Biobank. We replicated in AFGen, and we looked at the relationship between these genetic scores and the atrial fibrillation as reported in UK Biobank, and is about now 20,000 cases. So if you look at what we found, the QT to some extent is our negative control. When we look at this data all together, and Parag did it, this is the student who was in charge of this project, what you can see is that within the normal range, more or less, of the PR interval or P wave, I'm showing the PR interval, but the pattern is very similar. The longer the PR interval and the longer the P wave, the lower the risk of atrial fibrillation. And this is not what we expected, although, from the data that we had, it was comforting to see that this is also a possibility, right? That we have both long and short causing. So we have more than one type, if you like, of atrial fibrillation. And you can see that the QT interval is not really significantly associated when it is genetically determined. And so one could therefore conclude from here that we have this component here that might, that we know is more common in women and in young people, the association between a short P wave and PR interval and atrial fibrillation, whereas we have in patients with ischemic heart disease or heart failure, another type of association of a longer PR interval and atrial fibrillation, but we know that this component here is causal. So from here, I would like to then move to the second and last component of my lecture that is really related to cardioembolic stroke mostly, and the relationship between atrial fibrillation, the substrate of atrial fibrillation, and cardioembolic stroke. So my second hypothesis here is in humans, atrial fibrillation is always the consequence of an underlying cardiomyopathy, maybe subtle, but there is something wrong with the heart. And that underpins both the arrhythmia and the increase in stroke risk. And, this may be independently, right? So the arrhythmia will make things worse, but is the substrate that can cause potentially directly an increase in cardioembolic, in the risk of cardioembolic stroke. So how do we prove this? What is the evidence that this hypothesis might be true? Well, there is, to some extent, we have to move out of the way we think about things. I mean, certainly the way I thought about things, because this is how I thought about. I thought, okay, pulmonary vein, atrial fibrillation, remodeling, electrical structural re-entry, and and that is, the kind of bias that one has when one studies healthy animals induces atrial fibrillation and comes to the conclusion that atrial fibrillation must be the cause of all evil, because otherwise, we wouldn't have, this situation. But in fact, animal models very rarely get stroke or, so, there is something else going on. And in humans, I think it is much more likely that both genetic, long-term exposure to both genetic and environmental risk factors would contribute and collude, if you like, to generate many different atrial substrates depending on the combination of risk factor and the genetic makeup, that then may favor atrial fibrillation and possibly also stroke and a cardioembolic stroke. And then you have all of that we know. I mean, this is still there, but it is downstream an initial atrial substrate. So again, what evidence do we have that this is the case? Well, we have recently very interesting data from genetics, from very, very large cohorts, 60,000 cases of atrial fibrillation, more than 1 million people, suggesting that a large proportion of the gene variants that are really related to atrial fibrillation are known to affect the cardiac structure. So they're not just electrical, but they are also structural. Not only that, but and this has been described, very much for contractile protein, from structural protein in the myocardium. And in fact, even rare mutations identified in Iceland really pinpoint the direction to an effect on contractile myofilaments. And when that effect is on the structure, it is also associated with a higher risk of stroke. Then there is the lack of a strong temporal relationship between episodes of atrial fibrillation or presumed atrial fibrillation and cardioembolic or thromboembolic events. This has been shown by the ASSERT study a few years ago now. But again, really only 15% of the episodes of high atrial rate are preceding a thromboembolic event by less than 30 days. Otherwise, long or short that they can be either quite remote from the thromboembolic event, more than 30 days with a median of 6 months, or occur after the stroke. So that raises some doubts. And, the third, if you like, piece of circumstantial evidence that there may be something else going on here are the results of the COMPASS study, because in this study here, it was clearly demonstrated that low dose of rivaroxaban with or without aspirin associated is perfectly capable of reducing ischemic stroke risk, even in the absence of atrial fibrillation, but in high-risk patients, patients with stable cardiovascular ischemic heart disease. So what do we know? Or what do we think we know? Well, we know that we see a thrombus in the left atrial appendage. And we have for many years been aware of the Virchow triad, by which, stasis, blood flow abnormalities, a hypercoagulable state, endothelial cell dysfunction or injury, both of these you have in the context of inflammation, for instance, can't favor the formation of a thrombus. And there has been evidence that obviously with the fibrillating atrium not contracting efficiently, then, we will have that is associated with an increased risk of thromboembolic events. But, we couldn't really quite quantify or understand the blood flow abnormalities in the left atrium until recently, because now we can measure it quite precisely using MRI. And here is an example of how the flow in the left atrium of a healthy individual with a CHA2DS2-VASc score of 0 in sinus rhythm looks like. You have a very tight vortex anti-clockwise that really washes the walls of the atrium, and it's no surprise that under these circumstances you do not have thrombus formation, you know, and it really is very well designed to prevent that. So what happens in atrial fibrillation? Here you have a patient with a CHA2DS2-VASc score of 4 and persistent atrial fibrillation. And you can see there is some kind of vortex, but everything is slower and less organized. So what happens if I take these patients, I cardiovert it, and then I measure it again after 4 to 6 weeks, and you find that, okay, we're looking better, but not quite normal. You know, the velocities are lower, the vorticity is lower, and you know, you may say, well, you know, this is what happened with atrial fibrillation. You know, you have a sick atrium, and probably as a consequence of your atrial fibrillation, and that's why you are in this predicament. But actually, if I compare this with patients who never had atrial fibrillation, and you know, long-term monitoring has excluded atrial fibrillation, but have the same stroke risk factor, then the picture is exactly the same. So between these two categories, there is absolutely no difference in vorticity or in velocity or stasis or other parameters, you know, that you can see here some of the differences, you know, there are none. So does this in any way, is any way linked to stroke? And here, again, Marco, in another cohort of 134 patients, 44 with a history of large, presumably embolic or lacunar stroke, has found that the vorticity is really an independent risk, you know, is associated, let's say, you know, because this is not a prospective study, is associated with cardioembolic, large cardioembolic strokes in this cohort. Very much so, and independently of left atrial or left ventricular ejection fraction, left atrial volume, CHA2DS2-VASc score, or a history of atrial fibrillation. So, you know, the flow of the, you know, the 3D flow, blood flow in the left atrium here, and the ability to form a tight and fast vortex are very much, you know, this study suggests, linked to the pathogenesis of the left atrial thrombus. So that we believe it's important because it might help us to stratify who actually might benefit from anticoagulation, whether or not they have atrial fibrillation. And it also kind of opens up the idea, you know, if we only look at the atrium, we then call this an atrial myopathy. But if we look at the whole heart, then we can understand better why this is happening. And as you can see here, you know, the blood flow characteristics in terms of vorticity and velocities do not seem to be at all linked to ejection, to the left ventricular ejection fraction, which is, however, you know, not very low in these patients, is reduced but not very low. But it is very much linked to the left ventricular diastolic function. So there is, again, that kind of cardiomyopathy that we haven't quite understood sufficiently that is causing both. And, you know, again, a few years ago, we did demonstrate that as well in patients with mostly paroxysmal and lone. I know that we were not supposed to use lone, but just to say that there were no obvious comorbidity in these patients. And in this group, we did see a reduction in the phosphocreatine to ATP ratio by MRI, MRS, magnetic resonance spectroscopy, phosphorus, suggesting really that an energetic imbalance in these hearts. And you say, well, this is because they have atrial fibrillation and high rate and then whatever. But actually, not because after 9 months after a successful ablation, this abnormality remains, suggesting that it was there to start with. And the same is the case for blood flow, stress and blood flow. Exactly the same picture. Last 2 slides really looking at, we looked at that, blood flow abnormalities, and we can measure it. What about this other part? And here we have teamed up with one of my colleagues, Haris Antoniadis, who has had this very interesting data on the ability to deconvolute the CT signal of perivascular fat according to the molecular analysis of the fat itself using RI. So he calls that radiotranscriptomics. So you can image the fat, you can take this fat, perivascular fat out at the time of surgery. You can then measure a number of biomarkers in here and teach your system to recognize what patterns in the CT signal is connected with those signals. And by doing that, he has been able to really very much increase and refine risk stratifications of patients with ischemic heart disease, and even identify the holy grail of the vulnerable plaque. So applying the same technology to the periatrial fat and the atrial wall, we are, as you can see here, working and hoping to do the same. And we do that on a very large scale because this can be done on clinical CT scans. All the CT scans that are done in the context of chest pain, for instance, in the whole of the UK, and then link this to with passive follow-up to the UK NHS Digital, where all the events in these patients are stored over time. So again, results from this, I think, will be ready in less than a year. So in summary, what I hope I have been able to demonstrate is that studies of mechanisms that underpin the atrial fibrillation-induced remodeling really have uncovered novel, interesting pathways that regulate atrial repolarization and remodeling itself. Now, to which extent these mechanisms are actually causally related to the maintenance of recurrence of human atrial fibrillation, it remains to be fully established. But I believe that this trend to use multi-omics and imaging and Mendelian randomization studies will definitely help to answer this question in quite the near future. I am extremely enthusiastic about the advances in atrial imaging techniques and what can they bring to the field, because there is really a potential here to refine stroke risk, both in the presence and in the absence of atrial fibrillation, and target it better to the patient that need it most. The next step is obviously to assess whether these developments can allow us to practice that personalized medicine that is the ever elusive world, at least in the cardiovascular field. And I believe that, you know, this is pointing us in the right direction. So 2 final messages to the students that are in the audience. I mean, what has the pandemic taught us? It has taught us that we need to be flexible and we need to be adaptable and imaginative to stay afloat in a crisis. And this is an interesting article that was published in The Economist in October that shows that people who have a lot of external collaborations have done really well in the last year or so. You know, you can see the number of publications, but more importantly, the impact of those publications. They have done better than people who have a large group, because, you know, it is still groupthink is the thing that does stop progress. And so multiple collaborations have been the key for the success of the people who have managed to have and build that kind of network. And the other point that I want to make is related to this quote from an American chemist and president of Harvard University, and what I was saying before. You know, he says, behold the turtle. He makes progress only when he sticks his neck out. And sticking your neck out in English really means taking risks. And if there is something that one has to do in an academic career, is to take risks. Without taking risks, there is definitely no progress. If one takes risks, there might be progress, but, you know, without it, you're sure that you're not going really to progress. And with that, I conclude, and thanks this fabulous group of collaborators and the team member. You know, as you can see, there are very few British people in my lab and amongst my collaborators. Oxford is a wonderful international place and may remain that way after Brexit. Thank you very much for your attention.
Talk, very comprehensive. I guess there are many questions from the audience, so maybe we can start for the chat. Are there any questions? No? Okay, from the audience, who wants? Uli has a question.
Hi, Bauer. Can you see me?
Yes, yes, I can.
Yes, okay, all right. Thank you very much for this very, very impressive talk. And I'm very much intrigued, as you most likely are also, by the result of your Mendelian randomization study, which shows surprising results. Here in this case, that the longer PR interval is associated with a lower risk to AF, something that has not been shown at all before. And for the P wave, I think there was a comparable trend in the paper, which I can understand because we know that stronger repolarizing currents are associated with a higher conduction velocity, but also with more reentry, so we can understand that. The other part with the PR interval is more difficult to understand. And my question is whether you tried, maybe together with Blanchard, to check whether the genetic risk score you used for that contains variants that might potentially have an effect on other electrophysiological factors, maybe in a modeling approach.
Yeah, it's a very good question. You know, I get the echo again. Yeah, and that's better, perhaps. Yeah. So the ion currents, well, that is a jump. Let's say the variants that are associated with differences in the PR interval are very many. There have been many described. And unlike on the P wave duration, probably because it is easier to measure the PR interval than to measure accurately the P wave duration. So in some respect, the data on the PR interval are more robust. Now I am, I very much regret that I am not in Maastricht personally, because this is something that I would have liked to discuss. We think about conduction velocity and also the AV node for the PR interval. But I have a suspicion that there may be something else underlying the P wave duration of the PR interval. And I am, with Blanca, what I am trying to do is not so much to model the current, because one doesn't really know what these variants might cause. They are all around the sodium channels, but for instance, for the P wave, one of the strongest variants is on the SN10, channel that underpins the late sodium current, right? So not so much, we're looking at actual potential duration here. And the longer the P wave in that, associated with that variant, the lower is the risk of atrial fibrillation again. So I suspect that there is something else. And I am trying to get Blanca to work on this and try to think a little bit differently, and not only in terms of conduction velocity, but also on other characteristics. I am interested in the relationship between action potential duration and conduction, in both directions, for instance.
Yes, I think that is an important topic, certainly. But maybe we can talk about that on another occasion. Thanks.
Yeah. Any more questions? Well, I would like to go back to the microRNA, of course. Yeah, so I was just wondering if you looked, was there differential expression of the miR31 between the right and the left atria? And where was it actually expressed, in which cells?
Yeah, so it is expressed in myocytes, and it is expressed in fibroblasts. That for sure. In the increased expression, I mean, Svetlana has taken this work further and has produced a mouse with a fibroblast overexpression of microRNA 31 that also has an increased risk of atrial fibrillation inducibility. So, there are, it may well be involved both in electrical and structural remodeling in that respect. So you then said left and right, yes. It is expressed both in the left and right myocardium, but not in the ventricular myocardium of patients with atrial fibrillation, and also goats.
Is Svetlana looking also at other non-coding RNAs besides microRNAs, or she's focusing mainly on this one?
At the moment, she's looking, the data that she has are on the microRNA 31, in addition to other stuff that she's doing, but microRNA-wise, that is the mouse that she has recently made.
We have one more question, Kevin.
Barbara, thank you very much for this very nice presentation. My name is Kevin van Ooy. I'm a cardiologist in Maastricht. You can see
hi.
me? Yes, hi.
Yes, I can.
what for me is really striking, there's a big overlap between heart failure and atrial fibrillation. And for me, it's really striking when is the patient suffering from complaints for heart failure, when it's from atrial fibrillation, and especially how about measuring atrial function or atrial myopathy. That could be a missing link between both entities. And I was wondering what your vision is on the atrial myopathy as a link between heart failure and atrial fibrillation. Did you hear my question? No. Somehow connection is lost.
We have to reconnect. I think we have a small technical problem. 1 second, please.
Do you hear us now?
Yeah, I can hear.
Okay, so probably you missed completely the question.
I got the up to heart failure.
Oh yeah, so for me, I'm always in discussions with our heart failure physicians, especially on the overlap between atrial fibrillation and heart failure, especially also in HFpEF. When is the patient suffering from heart failure complaints, when is he suffering from atrial fibrillation? And somehow we're now trying to study more carefully the atrial function, atrial myopathy, as maybe a missing link in between. We have varying methods. Of course, we can have echo, we have now strain. Of course, ECG is helping us. We can measure voltage maps during the EP procedures to see if there's atrial myopathy. But still, it's quite hard to really understand atrial function in relation to atrial fibrillation and heart failure. And I was wondering what your vision is on atrial function in relation to heart failure.
I think, I think there is one cardiomyopathy that affects the left ventricle and the atrium. And so that one can really understand a lot about the atrium by looking at the ventricles. And I was very surprised recently. Uli has worked also in these areas, but we had finally managed to look at biomarkers of incident atrial fibrillation after cardiac surgery in patients with sinus rhythm. And we had, in collaboration with Roche Diagnostics, a number of biomarkers, and the idea was that there was inflammation essentially that was associated with atrial fibrillation after cardiac surgery. So we have this enormous panel of inflammatory markers, et cetera, et cetera, et cetera. And at the end, the only markers that really trumps them all, including echocardiographic measurements, is NT-proBNP. So NT-proBNP is a very strong independent predictor of incident atrial fibrillation in patients in sinus rhythm after cardiac surgery, when you would think that really the inflammation would play a very important role. So definitely, both the genetics, the biomarkers, and what we know point in the direction that there is a cardiomyopathic substrate in heart failure is, if you like, extreme, but is there all the time. You know, that is what I am convincing myself at the moment, because every time I look at something, it always points in that direction.
Okay, I guess there are no more questions. So to close or to finish, we would like me and Tilman to give you something. Of course, this would be much nicer if you were here, but nevertheless, we hope to welcome you in Maastricht sometime soon.
Shall we give it together?
Yes. So can you see it?
This is for you, Barbara. And we extend
Thank you very much.
We extend our invitation until next spring, and then we'll invite you again. And then you get the promised dinner in the castle, and maybe you can have a short visit to the TEFAF if Corona allows. But we will welcome you again. And then this is very heavy, so you have to check it in on your way back. But I hope that will not be a problem. It will be waiting for you. Thank you very much.
Thank you very much. And thank you not just for now, but for all the help and counsel and advice you have given me over the years. I'm very grateful. Yeah. Thank you.
Thank you. Bye-bye.
Hey, Paolo. Bye-bye.
So you can chat a little bit. I need a couple of minutes, but you have to stay here, please. So, you might ask yourself, what is this CARIM priori all about? You have seen it already in our new organogram, and I know by now you must notice that it has something to do with our daily board. So I will explain this now, and I hope you will understand and how we came across this new idea. Last summer I was on holiday, and I walked from Bologna to Florence, and obviously my geographical knowledge was way less than my peptide knowledge, because I forgot that there's these mountains between Bologna and Florence, the Apennines, and so we had like 5 days of walking, but also 5,000 meters of climbing, like all together. So we had to rest a little bit in Florence, and we took a couple of days, and there we noticed that this city, like already 500 years ago, had pretty modern views on governing. And then I brought back this idea to Wouter. I first thought that was ridiculous, but Wouter said this is the best idea since sliced bread. So then we developed this idea and now it comes alive today. So actually in Florence during the Renaissance there was like a couple of periods where the important family, the Medici, were absent in governing the city and the Republic. And at that time the city government consisted of three bodies as accounted for here. It's a signoria, which is basically comparable to our CARIM DB, and they were advised by two councils, the Collegi, the College of 12, the Dodici, and comparable actually to the CARIM Strategic Board, and also the Council 16, which is actually comparable, I believe, to our school council. And for every council, the representatives should be of good standing, so as they have to be within our CARIM organization as well. And the signoria was assembled, and now it's getting interesting, from 9 members of the guilds. And there were 21 guilds in Florence at the time, very comparable to our CARIM PI groups. And they were divided in 7 large guilds, 5 medium, and 9 small. It's also a bit comparable as CARIM, because we have very small PI groups and also very big ones, more influential and less influential. So it's basically all the same. But then the interesting stuff happens, because the members of the signoria, they were elected by a lottery, by a drawing. So the names of all guild members in good standing older than 30 years were put in a letter pouch from which the members of the signoria were pulled by a people's representative. And the signoria governed for 2 months. And we said, well, 2 months is a little bit too short, but we said 1 year might be nice. So we're going to elect additional daily board members from the CARIM employees, and they have a chair or a seat for a year. So let me explain the procedure. We're going to expand the CARIM Daily Board by drawing. And in our SAP download of September 2021, we had 597 CARIM employees. Well, this is very impressive. So many people have a connection, work with CARIM. From those, 302 are paid. So like the rest is mostly external PhD students that get their pay, their funding from different sources. And then we come to the fixed position, because as in Florence we have the guilds, you're mature, you have a fixed position at CARIM, and we have 113, and the CARIM Bureau is excluded in this draw. We have 71 males, we have 42 females, and within this population there are 79 scientific personnel and 34 of technical personnel. So it's open to everyone with a fixed position. So from these pouches, the little one is the female pouch, the slightly bigger one is the male pouch. There will be 3 names drawn, position 1, 2, and 3. And then, depending on acceptance of this board seat, so we'll ask number 1 first. If number 1 declines, we go to number 2, etc. So we have 6 possibilities for these 2 positions. Ideally, 1 male, 1 female. And then, as of January 1, if desired, those 2 can join the CARIM Daily Board with all their rights and duties that are coupled to this the governing structure. And then the names that are not used or that are not make it to the board, they'll put back in the bags, and there will be a next draw in our next annual symposium. So, as said, the DB priority membership runs from January 1st till 31 of December. And I would now like to invite Sophie to do the drawing.
please use a microphone for our members online.
Johanna Driessen.
So can you call them again?
Carla van der Kalle, Liedewij Bos en Johanna Driessen.
For the ones that are interested, these are the names that are in the bags.
Bastiaan de Gelaan. Wouter Verhezen. Paul Schiffers.
So the names have been drawn. So these are 6 potential new daily board members of CARIM. We will now contact those people individually and see if they accept the challenge. And then we'll decide and communicate with you who our 2 extra daily board members will be for the year 2022. Well, congratulations, at least candidates, and this will be continued. Thanks. Now there is a sort of additional session, and these are the prizes for the CARIM community. And we will start with HS BAFTA. And many of the recipients are grossly overdue in getting their nice medals as displayed here. But I think we should start with the recipients of this year, and that is 2021. And those names are here. So if you are here, please step forward and collect your medal, please. And afterwards, for all BAFTA recipients, there will be a picture on the stairs, like in the main hall. But you can, of course, make pictures here as well.
Please use a microphone.
And then we have the round of 2019-2020. And from 2019, please come forward. If you're here, Mohammed, Federica, Cengiz, Walid, Roger, Valeria, and Rob. especially for this guy in pink. He's like bugging me already for 2 years. And I just know he will take the golden one, because that's the bling bling. Yeah? Roger, what's your pick? You can take the stands as well, because you can display them. Hop, also the golden one. Congratulations, all 3 of you. And the rest, there will be some other ceremony at CARIM office. And then the round of 2020, that's even a longer shot. I think when some you can see that get them twice. So they get first the PhD award and then they get the postdoc award. So we decided that one of these nice BAFTA medals is enough. So if you are twice on the list, you better stay seated. So for 2020, please step forward if you are here and pick up your medal of choice. Okay, congratulations to you 3. And then the next award is the thesis award. So every year CARIM decides who had the best thesis in the preceding year. It's always a very difficult decision because our committee has to read many of these PhD theses, but in the end they always find a winner. And I have a jury report here. And the jury consists this year of Paul Volders, Eline Kooi, and Casper Schalkwijk, and I'd really like to thank them for the enormous job that they did again, and also in finding the winner, and also making this very nice jury report. But I won't keep you away now from the winner, and the winner is this year Martijn Smulders. Is he here? Yay! So it's a huge report, 6 pages, so you better take a seat. So the title of your thesis is Diagnostic Evaluation of Chest Pain, the Role of Non-Invasive Cardiac Imaging. And your supervisors and promoters were Harry Krijns and Joachim Dilberger, and your co-supervisors were Bas Bekkers and Bas Kietselaar. So first I would really like to congratulate you, and then I will read to you what the jury thinks of your thesis. Martijn started his scientific career as a medical student at the University of Maastricht. And he analyzed clinical and cardiac magnetic resonance data of patients who had suffered an acute myocardial infarction. His remarkable ambition and dedication led to successful co-authorships of several manuscripts. Immediately after his MD degree, he continued as a researcher at the Department of Cardiology. In 2015, he received a Netherlands Heart Foundation DECA grant, allowing him to set up the Carmenta trial, which you all know about. And this landmark trial on patients suffering from a non-ST elevation myocardial infarction showed that an early non-invasive imaging strategy, CT or CMR, was feasible and safe compared to an early invasive strategy. In 2017, Martijn won the 22nd Pellerin Prize of Maastricht UMC based on this project. The final results were published in the prestigious Journal of the American College of Cardiology in 2019 and later implemented in the 2020 ESC guidelines even, so that's a big achievement. During his PhD trajectory, Martijn was awarded twice, in 2016 and in 2019, with the Best Oral Presentation Prize at the Annual Congress of the Netherlands Society of Cardiology. And in 2020, he was nominated among the 3 best dissertations of the Eindhoven Dissertation Prizes of the Netherlands Society of Cardiology in this country. While he is currently finishing his cardiology training, his cardiology training, he shows clear ambition to combine clinical care with a research career at the junction of cardiology and cardiovascular imaging. With the CARIM Dissertation Award 2020, Martijn receives significant recognition that will help him to proceed as an academic cardiologist at Maastricht UMC+. That's it, from the jury. Congratulations.
Thank you.
Now we have to find the prize somewhere. I think it's in a box still. We have to hide it because everybody is peeking everywhere to see who the winners are. Here it is. It's also heavy, it's very nice. Thank you. And lastly, we come to the most important award within CARIM, and that is the CARIM Commitment Award. And this year's recipient is actually long overdue, and I was always hesitant to put her forward because she's so close to me. But this year we got many other recommendations as well, so I'm very happy to announce that the 2021 recipient of the CARIM Commitment Award is Stella Thomassen. I guess you are here, somewhere in the audience. Stella graduated I think in '89 at the Higher Laboratory School in Belgium. And then her mother thought, well, maybe she should get a job close to home. And she was trained in biochemistry. So your mother knew that there was a department of biochemistry at the Young University of Maastricht. So one day she took her handbag in one hand, it's called a sakosje in Belgium, and in the other hand she took Stella and she went to our biochemistry previous department chair, the famous biochemist and enzymologist Jan Rozing. And then she knocked on the door, she put her sakosje on the table and she told Jan, here's my daughter, she's going to work for you. And Jan had been in Jan had been in tighter spots, but I think he was so flabbergasted that he just said, yes, that's okay. Yeah, so Stella became a member of our Department of Biochemistry. And actually, I think you shaped our department. You have been here for then more than 30 years. And I actually wonder why the university missed that. Because officially, you're booked at starting at 2020, I heard today. So 2000. So 10 years is missing. So we don't know how that comes. But I think your anniversary will be corrected next year. So I think Stella always worked beyond her qualifications as a biochemistry technician. She's a fantastic researcher, but that doesn't merit the CARIM Commitment Award. But I do believe that your contributions, lab contributions, was pivotal in deciphering the very adverse effects of the third-generation oral contraceptives on thrombosis. And also, your work was pivotal in developing this anti-hemophilia drug together with Baxter, like 10 years later. But next to that, you are the best biochemistry member ever. I mean, your laughter, I mean, we always know when Stella's in, you laugh a lot, we hear you everywhere. As said, you shaped our department. You always organized everything in the lab. I mean, if we needed like the plasma pool, the normal plasma pool, before we actually needed it, you organized it. You did everything in blood drawing. You are this emergency response person, BHV'er. that like for all longest time. And also, you were like the engine, the motor, I would say, the V8 behind all social events in our department. I mean, whenever there was a party, you were there, you were organizing it, and you had the loudest voice, and you were heard everywhere. That's really good. Somebody that had never been in Stella's lab, you have to go there. This is the final year that you can visit it because we are moving. If you come in there and you look like this, it's a normal lab. If you look like that, you see 1,000 of beer bottles. They're like everywhere on the ceiling. And it's always collected from every trip we made. We had to bring beer back, we drank it, and the bottles were in the lab. They're actually making pictures of it now, but I think this is the last year. Yeah. The most original lab in the world. Yeah. So yet this enormous drive to make all these social things happen in our department, and we are very grateful for that. And also for the rest of the university and the UMC, everybody knows you. Wherever you come, like in all departments, they know Stella, and Stella knows everyone as well. Last year, a year from now, last year, you were hit with a horrible disease, and we know that. And this was a very heavy year for you. And whatever happened to you, you had so many therapies. You had chemo, you had radiation, you had surgery and chemo again. And whatever happened, you still had this positive mode. Everywhere, you were a fighter. And everywhere when you talk to you, and then you can say, well, sometimes you don't talk to me, and then I have a dip, I have this down day. But whenever you talk to you, you were actually giving hope to the other people, and even beyond the hope that you had yourself. I think you never lost hope. The only thing you lost is your hair. Yeah. And even that's coming back, I think, in a very cute way, especially this blonde little thing, which I always call you touched by an angel. And this is really what you are. So Stella, you're very dear to us. I hope you will stay with us forever. And it's from our heart, because you care, you get the CARIM Commitment Award today.
Shortly, I want to thank everybody who, all my dear colleagues who sent me flowers and cards and presents during this very difficult time. And I'm really glad that the heaven gates didn't want me yet. And I'm really glad that I'm standing here now in front of you. And I'm really thankful for this award. And I'm really thankful. And thank you very much, everybody.
And I believe this concludes our hybrid condensed CARIM Symposium 2021. I sincerely hope that next year we'll be up and running again, and we have this huge party again at Thyssen, which we so badly miss. But it will return, I will promise you that. If we only meet this 15% of vaccinations, I hope, in the coming year, or infection, that's also fine. Then our world will be saved. I shouldn't say this, but I believe it. Okay, so before we close, I would really like to thank Esther and Barbara for organizing this year. It's always like a huge task to do this. And please take a little roll outside or a piece of fly, there's still left. For the HS BAFTAs, there is a picture moment on the stairs. And for now, safe trip home. And I hope to see you tomorrow somewhere in our fine institute. So CARIM is the best institute in the world. We know it. I'm very happy that you all are a member. And see it as a playground to do everything you like. You can play your hobbies everywhere. CARIM will support you with any grant system or any other help. And I hope you have a great time with us. See you next year.